Welcome, fellow seekers of knowledge, to Vibing Science. Tonight, we embark on an extraordinary journey, not to the far reaches of the cosmos, but to a universe equally profound, the human heart. For centuries, love has been the muse of poets and philosophers, a mystery wrapped in emotion. But what if I told you that beneath the sonnets and sighs, there lies a breathtaking scientific ballet? What if love, that most human of experiences, is deeply rooted in the very biology that shapes our existence? Dim the lights, find your comfort, and prepare to have your understanding of connection, desire, and attachment reshaped. As we delve into the intricate dance of neurons and hormones, of evolution and experience, I invite you to settle in, open your mind, and if this voyage resonates with you, please consider giving us a like, sharing your thoughts in the comments, and subscribing for more profound explorations. Let's begin. Welcome, my friends, to a journey deep into the very core of what makes us human. Tonight, we embark on an exploration of one of the universe's most captivating and complex phenomena: love. Not the fleeting, superficial kind, but the profound, biological, and sometimes utterly bewildering force that shapes our lives. We'll peel back the layers, from the whispers of ancient evolution to the intricate dance of neurochemicals within our brains, all to understand that unseen architecture we call romantic love. For centuries, poets, philosophers, and artists have sought to capture its essence, to define its indefinable allure. Yet, as science advances, we find ourselves with new tools, new perspectives, to examine this elusive state of being. The journey into the biology of romantic love is still in its nascent stages, a relatively young field, truly. As of the year 2021, a mere 42 biological studies had dared to probe its mysteries. Think of it: countless volumes of poetry, endless songs, millennia of human experience, and only a handful of scientific inquiries. This speaks volumes about its complexity, but also about the incredible frontier of discovery that lies before us. The very term romantic love itself has been a shapeshifter throughout history. In its initial coinage, it evoked images of courtly love, tales of chivalry, and idealized, often unconsummated, affections. But in the halls of academic psychology and, more recently, biology, the definition has shifted. Here, we delve into a concept that transcends mere cultural constructs. We are looking at a brain system, or rather, a constellation of systems intricately linked to the fundamental human drives of pair bonding and mating. It's a biological imperative, a symphony orchestrated by our very physiology. In 2021, the comprehensive review by Bode and Kushnick offered a crucial biological definition, a compass for our journey. They proposed that romantic love is, at its heart, a motivational state. It's a profound yearning, typically for long-term mating with a specific individual. This state, they argue, manifests across our entire lifespan, from the first flush of youth to the twilight years. And crucially, it's associated with a distinct, measurable array of cognitive, emotional, behavioral, social, genetic, neural, and endocrine activities in both men and women. Throughout much of our lives, it serves vital functions, guiding mate choice, initiating courtship, facilitating sexual intimacy, and cementing the bonds of a pair. It is in essence a complex suite of adaptations and byproducts that emerge somewhere along the recent evolutionary timeline of our species. Now, it's vital to understand that romantic love, in this biological sense, isn't always confined to the tidy parameters of a reciprocal, social, or interpersonal relationship. You see, this powerful motivational state can be experienced even outside the context of an established connection. Consider the phenomenon of unrequited love, where feelings are poured forth but never returned. The pain, the longing, the obsession, these are all manifestations of this intricate biological system, even when it's one-sided. A person can harbor profound romantic feelings for a potential partner before any real relationship has even begun, even for someone they barely know or are not acquainted with at all. Think of love at first sight, or the deeply personal, often intense connections we form with public figures or fictional characters, what scientists call parasocial attachments. These are not mere whims. They are echoes of this ancient system at play. The early stages of romantic love, that dizzying period we often refer to as being in love, or infatuation, limerence, or passionate love, frequently carry an obsessive and almost addictive quality. It's a whirlwind, a tempest of emotions and thoughts. However, it's important to differentiate this early, often intense phase from what attachment theorists like John Bowlby described as the attachment system. For a time, some argue that attachment theory could encompass all aspects of love, but modern researchers of love now understand that romantic love and attachment, while deeply intertwined, are not identical. The initial surge of romantic love, with its unique blend of exhilaration and sometimes consuming preoccupation, seems to involve additional brain systems, each with its own distinct evolutionary lineage, serving purposes beyond mere attachment. And let's be clear, romantic love, while often accompanied by sexual attraction, is also distinct from it. Though they frequently occur together, they are not the same. The way romantic love manifests can vary wildly from person to person. A fascinating cross-cultural study of individuals currently in love identified 4 distinct clusters, each characterized by differing degrees of intensity, obsessive thinking, commitment, frequency of sexual intimacy, and other nuances. Other research indicates that romantic love can be experienced both with and without those intense, obsessive features we often associate with it. Typically, this intense romantic love is thought to have a limited lifespan, lasting perhaps 12 to 18 months, or at most, around 3 years. However, there's a rare and intriguing phenomenon known as long-term intense romantic love, where some individuals maintain that fiery, almost early-stage attraction within a relationship for 10 years or even more. The key difference here is a notable reduction in those obsessive characteristics that define the initial heady rush. Neuroscientists, peering into the very fabric of our being, increasingly believe that our basic emotions arise from distinct, specialized circuits within a brain. Many of these primary emotion motivation circuits are shared with other mammals, a testament to our shared evolutionary heritage. These ancient brain systems evolved to direct behavior, to guide us through the fundamental imperatives of survival and reproduction. Among these primary neural systems, researchers hypothesize are at least 3 discrete, yet interrelated emotion motivation systems in the mammalian brain, specifically designed for mating, reproduction, and parenting. Helen Fisher and her colleagues, pioneers in this field, have eloquently described these as lust, attraction, and attachment. Lust, in its simplest form, is the sex drive, the raw, primal libido that propels us towards sexual activity. It is the biological engine of procreation. Attraction, which we can equate with early-stage romantic love, or passionate love, or infatuation, is the exhilarating current that sparks between individuals. It's associated with feelings of intense joy, with those relentless, intrusive thoughts about the beloved, and with a profound craving for emotional union. It's the magnetic pull that draws us together. And then there is attachment. This is the attachment system as theorized by John Bowlby, often referred to as companionate love. It's the deep, enduring bond associated with feelings of calm, security, and comfort when near, and a profound sense of separation anxiety when apart. It's the anchor that keeps us connected through the storms of life. In Fisher's model, these systems, while tending to act in concert, can also become disassociated, operating independently. Imagine this. A person in a long-term partnership might feel a profound sense of attachment to their spouse, the bedrock of their life. Yet, simultaneously, they might experience intense romantic love or attraction for someone else, while also feeling a purely sexual attraction or lust for still others, all three operating within the same individual at the same time. This decoupling, this independent functionality, can also explain phenomena like platonic infatuation, where an individual falls deeply in love without any accompanying sexual desire, as explored by Lisa Diamond. Fisher initially associated each system with distinct neurotransmitters and hormones: lust with estrogen and androgens, attraction with dopamine, norepinephrine, and serotonin, and attachment with oxytocin and vasopressin. However, as modern research deepens, we understand that these associations are not quite as clearly compartmentalized as initially proposed. The beautiful complexity of the brain means that romantic love has also been linked to endogenous opioids, cortisol, and nerve growth factor, elements not included in Fisher's earlier framework. Furthermore, the idea of completely independent systems has evolved. A more contemporary view suggests that the attachment system is, in fact, active even in the early, infatuated stages of romantic love, intertwined with the infatuation component. While Fisher's model, though groundbreaking in its time, is now considered somewhat outdated, the fundamental idea of these interrelated systems remains incredibly useful as we unravel the intricate tapestry of love. Now, let us turn our gaze to the grand stage of evolution to understand why these systems, this profound phenomenon of romantic love, might have come into being. Evolutionary psychology, a powerful organizing framework, offers compelling explanations for psychological functions, moving beyond mere description to understanding their adaptive purpose. It posits that a given trait must, in some form, provide a reproductive benefit to an individual. From this lens, romantic love emerges as a remarkably potent commitment device. Think about it. Romantic love powerfully suppresses the search for alternative mates. It can do so with an almost irrational intensity, making us blind to even more desirable potential partners who might cross our paths. This singular focus not only solidifies the bond with our chosen partner, but also signals that commitment to them. More subtly, it can also signal to potential alternative mates, subtly discouraging their pursuit. The emergence of longer pair bonds in human evolution coincided with a fascinating biological shift: concealed ovulation. Unlike many other species, it became impossible to generally determine when a human woman was ovulating. This biological riddle necessitated partners staying together, engaging in sexual activity throughout the entire menstrual cycle to maximize reproductive success. Commitment, therefore, became an adaptive strategy, facilitating this continuous bond and, crucially, enabling the prolonged and demanding care of human offspring. The exhilarating, sometimes bewildering feelings of love, then, might be the psychological reward our brains produce when the complex problem of commitment is being successfully solved. The sheer intensity of romantic love, the way it can make people seem to become fools for love, can be understood through the lens of the handicap principle. This principle addresses the eternal contention between honest and deceptive signaling in nature. When true emotions evolved, a niche would have opened for sham emotions, for less risky, superficial displays. But how can honest signals persist without being devalued by fakers? The answer lies in expense. An honest signal can evolve if it's simply too costly to fake. Consider the peacock's tail, a cumbersome, nutrient-draining display. Only a truly healthy, robust peacock can afford such a magnificent burden. Females, therefore, use it as an honest indicator of health. In a similar vein, romantic love, with its bewildering, besotting intensity, its tendency to make us act like handcuffing oneself to railroad tracks, might have evolved as a handicap. A costly, yet utterly honest, display meant to prove the absolute authenticity of one's commitment. Romantic love may also have evolved to override rationality itself, to propel us towards reproduction, regardless of the considerable costs of raising a child, and despite any rational inclination to remain single or child-free. It serves as a powerful, almost instinctive counterargument to purely logical considerations. Furthermore, romantic love signals paternal investment. The involvement of a father in the form of stable pair bonds has been consistently linked to better outcomes for children, both as infants and throughout their development. Children raised within such bonds tend to be more socially competitive and significantly more likely to survive to reproductive age. The feelings of love, then, become a psychological mechanism that fosters this critical parental investment. Being in love can also spark creativity. This suggests that romantic love may have evolved, in part, as a sophisticated courtship display. Art, music, literature, these profound expressions of the human spirit, might serve a function akin to the peacock's tail, but as a dazzling display of mental prowess, designed to impress and captivate a potential partner. Some authors even suggest that creativity, particularly in this context, has historically been a significant component of the male courtship display. From a pragmatic evolutionary perspective, romantic love can conserve both time and metabolic energy. By focusing courtship efforts on a single, specific individual, it streamlines the complex and often exhaustive process of mate selection, preventing a wasteful scattering of resources. And the benefits extend beyond reproduction. Successful pair bonding predicts better health and survival. Happy, well-functioning romantic relationships contribute significantly to both mental and physical well-being, especially when navigating the inevitable stresses of life. Conversely, the dissolution of a pair bond, such as through divorce, is associated with increased vulnerability to disease, depression, substance abuse, and negative outcomes for children. Studies have even shown that victims of a heart attack are more likely to suffer another if they live alone. Love, in this context, becomes a vital ingredient for longevity and resilience. Monogamous pair bonding also plays a crucial role in preventing sexually transmitted diseases, which can severely compromise fertility, particularly for women. Certain STDs, like syphilis, dramatically increase the risk of miscarriage and can harm or be passed to an unborn child. The strongest predictor of contracting an STD is the number of sexual partners. Therefore, limiting this number through exclusive pair bonding is one of the most effective ways to safeguard reproductive health. Finally, romantic love promotes exclusivity through a powerful mechanism: mate guarding. Romantic jealousy, often considered one of the most common correlates of being in love, is an evolved protection against the perceived threat of losing one's beloved to a romantic rival. While jealousy, when it motivates us to maintain a healthy relationship, can be adaptive up to a point, it can also veer into pathological territory, where delusional or paranoid beliefs about infidelity take root, regardless of actual evidence. The precise moment romantic love evolved in human history remains a fascinating mystery, a subject of ongoing debate. Most theories, however, place its emergence either during or after the time of bipedalism. The earliest hominid with substantial evidence of bipedalism and some intriguing hints of pair bonding is Ardipithecus ramidus, dating back approximately 4.4 million years. Though it's possible bipedalism itself is even older. One compelling hypothesis suggests that monogamous pair bonding, a rarity among mammals, evolved during this era because walking upright required mothers to carry their infants in their arms or on their hips rather than on their backs. With their hands occupied, mothers would have been more vulnerable, necessitating additional support for food and protection from males of the species, the nascent husbands and fathers. Another significant selection pressure proposed is the evolution of infant altriciality, the extreme immaturity and helplessness of human infants at birth, coupled with a dramatically larger brain size. This occurred around 2 million years ago. At this critical juncture, the human brain became so large that a fully developed infant's head could no longer fit through the mother's pelvic birth canal, a challenge known as the obstetric dilemma. This demanded that infants be born much earlier and in a far more underdeveloped state compared to other species. This would have placed an even greater burden on mothers, making paternal support and a stable pair bond even more invaluable for the survival of their fragile offspring. Yet, due to the general scarcity of direct evidence from deep time, it's also entirely possible that romantic love, or at least a foundational precursor to it, predated both bipedalism and altriciality, potentially originating in a common ancestor of humans and chimpanzees, perhaps 5 to 8 million years ago. While chimpanzees primarily engage in opportunistic mating, some of their rarer reproductive strategies do exhibit features reminiscent of romantic love, involving mate guarding and more than fleeting associations. One assumption underlying hypotheses based on fossil evidence is that less sexual dimorphism in body mass, meaning greater similarity between male and female body sizes, indicates monogamy. However, this comparative similarity in human body mass only emerged as recently as 500,000 years ago. This suggests a multi-step evolution of human pair bonding, meaning romantic love could have developed during any of these protracted periods. The story, as you can see, is not a simple, linear narrative. Helen Fisher's influential theory posits that romantic love, which she considers distinct from attachment, functions as a powerful motivation system for selecting and focusing energy on a preferred mating partner. She believes this brain system evolved from what she terms mammalian mate choice, or courtship attraction. This phenomenon, observed across species, involved choosing a preferred partner based on displays of physical traits, like the peacock's dazzling tail or other distinctive behaviors. Fisher extends this to humans, incorporating the attraction to personality traits and other characteristics in her mate choice theory. Courtship attraction shares behavioral similarities with human romantic love, notably involving the activation of our dopaminergic reward circuits. In most species, courtship attraction is brief, lasting minutes, hours, days, or weeks. But in humans, intense romantic love can endure far longer. Fisher theorizes that as human pair bonding evolved, mammalian courtship attraction became prolonged and intensified, ultimately blossoming into the phenomenon we now call romantic love. However, a critique of Fisher's theory by Adam Bode suggests that courtship attraction primarily encompasses the initial spark of love at first sight, or a fleeting crush. Bode argues that the core components of romantic love, including intense attraction, obsessive thoughts, and attachment, actually evolved through a process called co-option, drawing from the ancient mechanisms of mother-infant bonding. A study on love at first sight reinforces this distinction. While individuals recalling such an experience retrospectively often describe features akin to passionate love, like constant thoughts and a strong desire for the person, those reporting love at first sight immediately after meeting someone new exhibited only neutral scores on measures of romantic passion. This led researchers to conclude that love at first sight is more accurately a strong initial attraction, rather than the fully fledged state of being in love. It more closely resembles courtship attraction. Bode, therefore, views courtship attraction as a separate system from the core components of romantic love. It may involve dopamine, oxytocin, and opioid activity, but much about it remains unknown as existing studies were not specifically designed to isolate and target this distinct phase. This brings us to the profound and fascinating concept of co-option. In evolutionary biology, co-option is a process where a pre-existing trait or mechanism is repurposed to take on an entirely new function. Think of how certain fish species, like catfish, co-opted their gas bladders, originally for buoyancy, to produce sound. Co-opted traits can be morphological, affecting physical form, or behavioral. This process can explain how a species develops a seemingly new evolutionary adaptation with surprising speed, sometimes faster than classical Darwinian gradualism might suggest. The structure already exists, merely needing modification to function in a novel way. In some instances, it involves altering the function of a single gene, while in others, a duplicate gene is co-opted, preserving the original gene's function. The terms co-option and exaptation are closely related, with exaptation emphasizing the structural continuity when a trait acquires a new use. Adam Bode has proposed that romantic love is not a single, monolithic adaptation, but rather a suite of adaptations and byproducts, a collection of interrelated systems. Crucially, several of these systems, specifically the attraction for bonding, the obsessive thinking, and the deep attachment, evolved by co-opting the ancient, powerful mechanisms of mother-infant bonding. This co-option theory suggests that the genes regulating mother-infant bonding were essentially duplicated and then repurposed to serve this new function of adult romantic love. Courtship attraction and sexual desire, while causally linked adjuncts that were combined and modified within romantic love, were not themselves co-opted from the mother-infant bond. This theory is supported by extensive human evidence, but also by a rich body of research on prairie voles, those fascinatingly monogamous rodents, which clearly demonstrates that their pair bonding utilizes the very same neural mechanisms as mother-infant bonding. The academic literature has drawn parallels between romantic love and the mother-infant dyad since the 1980s. Attachment theorists like Cindy Hazan and Philip Shaver-Long posited a shared biological process. In 1999, James Leckman and Linda Mays conducted a comparative analysis of early parental love and romantic love, revealing substantial similarities. Both are altered mental states, characterized by intense preoccupation, an exclusive focus on the other, a profound longing for reciprocity, and an almost idealized perception of the beloved. The developmental trajectories also align. Preoccupation intensifies during courtship in romantic love and around the time of birth in parental love, before gradually diminishing once a stable relationship is established or shortly after the postpartum period. And here's an uncanny similarity: the use of baby talk between romantic lovers. In 2004, Andreas Bartels and Samir Zeki made a groundbreaking leap, using fMRI to directly compare romantic love and maternal love. They focused on brain areas known to possess high densities of receptors for the attachment hormones oxytocin and vasopressin. Their findings were remarkable: precise overlap in specific regions, including parts of the striatum, the putamen, globus pallidus, and caudate nucleus, and some overlap in the ventral tegmental area. These are areas rich in dopamine and oxytocin receptors. Each type of love, however, also showed unique activations. Notably, maternal love engaged the periaqueductal gray matter, a region associated with endogenous pain suppression during intense emotional experiences like childbirth. More recent meta-analyses of fMRI experiments have further solidified these fascinating similarities between maternal and romantic love. A 2022 meta-analysis by Shi and colleagues found both types of love activated the left ventral tegmental area, a region more associated with the pleasurable, liking aspect of reward. Additionally, romantic love specifically involved the right ventral tegmental area, which is more linked to reward, wanting. In 2003, Lisa Diamond expanded on this idea, suggesting that adult pair bonding is an exaptation of the affectional bond between infants and caregivers. This ingenious concept helps explain phenomena like romantic friendships, platonic infatuations, and even passionate, deeply felt connections devoid of sexual desire. Dorothy Tenev, in her pioneering study of limerence, a state of obsessive, infatuated love, often for an unreachable person, documented instances where heterosexual women experience this type of intense, non-sexual reaction towards older women. Other historical examples include schoolgirls falling violently in love with each other, suffering all the pangs of unrequited attachment, desperate jealousy, etc., historically termed as smash, and accounts of Native American men who seem to fall in love and form intense, non-sexual bonds. Helen Fisher's theory, separating sexual desire from romantic love and attachment, further supports this. Diamond argues that romantic love, without sexual desire, can even transcend one's sexual orientation. It wouldn't have been adaptive for a parent to only bond with an opposite-sex child, so these systems must have evolved independently of sexual orientation. While sexual desire often initiates romantic love, Diamond suggests that shared time and physical touch can serve as powerful substitutes. She believes the connection between romantic love and sexual desire is bidirectional, with either capable of triggering the other due to shared oxytocin pathways in the brain. Given the evolving understanding of evolutionary theories and the limitations of Fisher's earlier neurochemical model, Adam Bode has proposed a more nuanced framework outlining 5 distinct yet integrated systems that coalesce into the phenomenon of romantic love. First, sexual desire, the primal drive to initiate and be receptive to sexual activity. A complex interplay of testosterone, dopamine, serotonin, norepinephrine, acetylcholine, histamine, and opioids are implicated in this fundamental behavior. Second, courtship attraction, which serves to choose and focus energy on a preferred mating partner, thus promoting courtship behaviors. This can manifest as love at first sight or a crush, and while it often precedes a relationship, it isn't a universal prerequisite. Dopamine, oxytocin, and opioids likely play roles here. Third, bonding attraction, the very essence of pair bond formation. It's characterized by a powerful desire for proximity, intense separation anxiety when apart, an exclusive focus on the beloved, and a heightened awareness of their presence. This is strongly associated with dopamine and oxytocin activity, particularly in the ventral tegmental area. Bode argues this is the type of romantic attraction observed in fMRI experiments of early-stage romantic love. Fourth, obsessive thinking, the relentless preoccupation or intrusive thoughts about the loved one. Some researchers have drawn parallels between this and obsessive-compulsive disorder, suggesting shared neurobiology, though evidence remains ambiguous. And finally, attachment, the system for pair bond maintenance, for sustaining those deeply close personal relationships. Its psychological features include a heightened sense of responsibility, a profound longing for reciprocity, and a powerful sense of empathy. Attachment is intricately linked to oxytocin, dopamine, and opioid activity, with some evidence also pointing to the involvement of vasopressin. Bode suggests that bonding attraction, obsessive thinking, and attachment, the 3 systems co-opted for mother-infant bonding, together form the essential core, the necessary components of romantic love. However, he emphasizes that all 5 systems merge, intertwining into the singular, multifaceted phenomenon we recognize as romantic love, with the precise blend and outcome varying wildly depending on individual circumstances and the dynamic of the relationship. It's a symphony of systems, each contributing its unique melody to the grand composition of human connection. Next, we will delve deeper into the intricate mechanics of love, exploring how reward, motivation, and even addiction shape our most profound connections. Welcome back, Cosmic Navigators. In our last journey, we began to peel back the layers of that most elusive of human experiences, romantic love. We acknowledged its multifaceted nature, a phenomenon so deeply interwoven with our individual and collective histories that its very definition shifts like the sands of time. But as we transition from its cultural definitions, we must now delve into the unseen architects of this profound emotion. Tonight, we're venturing into the ancient, silent workshops of evolution, where the very blueprints of love were first drawn. This is love's ancient code, an evolutionary blueprint. Imagine, if you will, the vast tapestry of life stretching back through epochs, billions of years of trial and error, of adaptation and survival. Within this grand design, every whisper of a breeze, every glint of sunlight on a leaf, every beating heart is a testament to the relentless, ingenious process of evolution. And deep within this intricate dance, a peculiar, yet powerful mechanism began to emerge, one that would bind individuals, drive reproduction, and ultimately shape the very trajectory of our species, the biology of romantic love. For centuries, philosophers and poets alike have pondered love's origins, often attributing it to divine intervention or an unexplainable magic. But modern biological sciences, evolutionary psychology, evolutionary biology, anthropology, and neuroscience, are now offering us a different narrative, one of neurochemicals and hormones, of intricate brain systems that produce the psychological experience and behaviors we recognize as romantic love. While this field of study, in its biological sense, is still in its nascent stages, with a mere 42 biological studies on romantic love as of 2021, the insights gained are already profoundly shifting our understanding. The initial challenge, as we explored, is truly defining romantic love from a biological perspective. Historically, the term was entangled with concepts like courtly love, laden with societal expectations and literary tropes. But for the biologist, the neuroscientist, the anthropologist, romantic love refers to a fundamental brain system, or perhaps a constellation of systems, intrinsically linked to pair bonding and mating. It's a primal directive cloaked in complex emotions. In 2021, a comprehensive review by Bode and Kushnick offered a working biological definition, a foundation upon which we can build our understanding. They posited that romantic love is a motivational state, typically associated with a desire for long-term mating with a specific individual. It's a phenomenon that spans our entire lifespan, manifesting in distinctive cognitive, emotional, behavioral, social, genetic, neural, and endocrine activities in both sexes. Throughout much of our lives, it serves vital functions: mate choice, courtship, sex, and, crucially, pair bonding. They propose a suite of adaptations and byproducts arising at some point in our recent evolutionary history. This biological definition is particularly fascinating because it detaches romantic love from the necessity of a reciprocated relationship. You see, romantic love isn't exclusively dyadic, social, or interpersonal in this scientific lens, even though it's undeniably linked to pair bonding. One can experience romantic love outside the context of an established relationship. Consider the pangs of unrequited love where feelings are not returned, or the sudden, potent stirrings a person might feel for a potential partner they barely know, a phenomenon sometimes called love at first sight or parasocial attachments. The emotional blueprint, the motivational state, can ignite even before a relationship truly blossoms. The early stages of romantic love, often characterized by an almost obsessive intensity, are frequently referred to as being in love, passionate love, infatuation, or limerence. It's a period marked by an intoxicating cocktail of emotions and behaviors, sometimes bordering on the compulsive. While research continues to refine its terminology, it's important to distinguish this early, fiery stage from the broader attachment system theorized by figures like John Bowlby. While these systems are deeply intertwined, academics increasingly argue that romantic love and attachment are distinct, albeit overlapping, concepts with their own unique evolutionary histories and neural underpinnings. Romantic love, in this sense, is also separate from mere sexual attraction, though they are often and powerfully experienced in conjunction. The expression of romantic love is, of course, as varied as humanity itself. Cross-cultural studies of individuals currently in love reveal clusters with varying degrees of intensity, obsessive thinking, commitment, and frequency of sexual activity. Some experience intense romantic love with pronounced obsessional features, while others do not. Typically, this initial, fervent intensity is thought to last between 12 to 18 months or up to 3 years. However, in a rare and intriguing phenomenon known as long-term intense romantic love, some individuals maintain that initial passionate attraction for a decade or even longer, albeit with fewer of the obsessive features that mark the earliest stages. It suggests a remarkable plasticity in our emotional wiring. Neuroscientist Helen Fisher and her colleagues have proposed a compelling framework for understanding the independent emotion systems involved in mammalian reproduction. They posit at least 3 discrete, yet interrelated emotion motivation systems within the mammalian brain: lust, attraction, and attachment. Lust, or the sex drive, is the primal urge, the fundamental engine of procreation. It's driven by hormones like estrogen and androgens. Attraction, often synonymous with early-stage romantic love, infatuation, or passionate love, is the intoxicating blend of exhilaration, obsessive thoughts, and an intense craving for emotional union. This is where the brain's reward system truly lights up, an arena we'll explore in much more detail shortly. Finally, attachment, also known as companionate love, is the bedrock of long-term bonds. It's associated with feelings of calm, security, and comfort, but also a distinct separation anxiety when apart. Fisher's genius lies in recognizing that while these systems often operate in unison, they can, crucially, become disassociated and act independently. Consider the individual in a long-term, loving partnership who feels a deep, abiding attachment to their spouse, yet simultaneously experiences intense romantic attraction for another individual, while perhaps feeling sexual desire for still others. This multi-layered emotional landscape is a testament to the distinct, yet interacting, nature of these systems. Lisa Diamond has even utilized this independent emotions theory to explain phenomena like platonic infatuation, where a deep, romantic connection can form without sexual desire. While Fisher's initial model associated each system with specific neurotransmitters and hormones, lust with estrogen and androgens, attraction with dopamine, norepinephrine, and serotonin, and attachment with oxytocin and vasopressin, modern research reveals a more complex, nuanced picture. These associations are not always as clearly delineated as first proposed. Indeed, romantic love has also been linked to endogenous opioids, cortisol, and nerve growth factor, components not initially included in Fisher's earlier framework. Furthermore, contemporary theories suggest that the attachment system is active even in the early stages of romantic love, not just in later, established relationships. While Fisher's model has evolved, her foundational idea of interrelated systems remains profoundly useful in our understanding. So, how did these intricate systems evolve? This brings us to the fascinating realm of evolutionary psychology, a framework that seeks to explain psychological functions not merely by describing them, but by tracing their adaptive origins, requiring that a given trait provide a reproductive benefit. Evolutionary psychology offers a rich tapestry of explanations for the emergence of romantic love. One compelling theory posits romantic love as a powerful commitment device. Imagine a world where mates are abundant and choices are limitless. Romantic love, in this view, evolved to suppress the relentless search for alternatives, even, perhaps irrationally, when a seemingly more desirable partner might appear. It acts as an internal signal of unwavering commitment to one's chosen mate, and perhaps, importantly, signals this commitment to potential rivals, effectively disincentivizing them from pursuing the individual. The emergence of longer pair bonds in human evolution coincided with another crucial development: concealed ovulation. Unlike many other species where ovulation is overtly signaled, in humans, it's generally not discernible when a woman is ovulating. This evolutionary shift necessitated partners staying together, engaging in sexual activity throughout the entire menstrual cycle to maximize reproductive success. Commitment, therefore, became highly adaptive, facilitating this continuous pairing and, vitally, supporting the demanding task of childcare. From this perspective, the intense feelings of love might be the brain's psychological reward, a pleasurable feedback loop when the complex problem of commitment is successfully navigated. But why the intensity? Why do people become fools for love, acting in ways that defy conventional rationality? This can be explained through the handicap principle. This principle, observed widely in nature, suggests that honest signals of quality or commitment must be costly to produce, too expensive for a faker to mimic. Think of the peacock's magnificent, yet cumbersome, tail. Only a truly healthy, Genetically fit peacock can afford the metabolic cost of such a display. It's a handicap. Yes, but precisely because it's a handicap, it signals genuine quality to potential mates. Romantic love, with its often bewildering, all-consuming nature, like handcuffing oneself to railroad tracks, as one author put it, may have evolved as such a handicap. Its very bewitchment, its besotting power, serves as an honest, expensive signal that one's commitment is truly profound, truly real. Romantic love, some propose, might also have evolved to override rationality itself. Raising a child is an enormous undertaking, fraught with considerable costs and sacrifices. Love, in this view, is a powerful psychological mechanism that compels us to reproduce, to embrace the monumental task of parenthood, even when our rational minds might otherwise lean towards remaining single or child-free. It's a biological imperative, disguised as a passionate yearning. Furthermore, romantic love strongly signals paternal investment. Research consistently links paternal investment in the form of stable pair bonds to better outcomes for children, from infancy through adulthood. Children raised within these committed partnerships tend to be more socially competitive and significantly more likely to survive to reproductive age. The very act of falling in love, therefore, could be an evolutionary mechanism ensuring the robust, long-term care essential for our offspring's success. The creative surge often experienced when in love, the sudden desire to compose poetry, write songs, or create art, suggests another evolutionary purpose: romantic love as a courtship display. Like the peacock's tail, artistic and intellectual expressions may serve as a demonstration of mental prowess, designed to impress and captivate a potential partner. Some even argue that creativity, in this context, is particularly a part of the male courtship display, a powerful signal of fitness and desirability. From a pragmatic standpoint, romantic love also serves to conserve time and metabolic energy. By focusing our courtship efforts intensely on a specific individual, it prevents the exhaustive, inefficient scattering of resources across multiple potential mates. It's a highly efficient targeting system for reproductive success. Beyond reproduction, successful pair bonding itself predicts better health and survival. Happy, well-functioning romantic relationships demonstrably contribute to both mental and physical well-being, especially when individuals face stress. Conversely, the dissolution of a pair bond, such as divorce, is associated with increased vulnerability to disease, depression, substance abuse, and negative outcomes for children. Consider the stark statistic: victims of a heart attack are more likely to experience another when they live alone. Love, in this sense, is quite literally a life-sustaining force. Monogamous pair bonding also plays a crucial role in preventing sexually transmitted diseases, which can severely compromise fertility, particularly for women. Certain STDs increase the risk of miscarriage that could be passed to an unborn child. Since the strongest predictor of contracting an STD is the number of sexual partners, limiting this number through committed pair bonds is a powerful evolutionary strategy for safeguarding reproductive health. Finally, romantic love actively promotes exclusivity through mate guarding. Romantic jealousy, a pervasive and often intense emotion, is one of the most common correlates of being in love. It's thought to have evolved as a protective mechanism against the threat of losing one's beloved to a rival. While adaptive when it motivates us to maintain our relationships, jealousy can, of course, spiral into pathological forms, manifesting as delusional or paranoid beliefs in a partner's infidelity, regardless of actual evidence. The precise moment during human evolution when modern romantic love first emerged remains a topic of scientific debate, veiled in the mists of deep time. However, the prevailing theories place its evolution either during or after the advent of bipedalism, the ability to walk upright on two legs. The earliest hominid with extensive evidence of bipedalism and some indications of pair bonding is Ardipithecus ramidus, dating back approximately 4.4 million years. One compelling hypothesis suggests that monogamous pair bonding, a rarity among mammals, evolved around this time because bipedal locomotion demanded mothers carry their infants in their arms or on their hips rather than on their backs. With their hands occupied, mothers would have been significantly more vulnerable, requiring increased assistance from males of the species, proto-husbands or fathers, for food and protection. This selective pressure could have driven the evolution of a stronger emotional bond, a nascent form of romantic love. A different, yet complementary, selection pressure is the evolution of infant altriciality and large brain size at birth, which occurred roughly 2 million years ago. At this critical juncture, the human infant's head became so large that it could no longer fit through the mother's pelvic birth canal if fully developed, a phenomenon known as the obstetric dilemma. This necessitated infants being born prematurely, in an underdeveloped and helpless state compared to other species. This profound helplessness dramatically increased the burden on mothers, making paternal support even more invaluable for the survival and thriving of their offspring. Yet, despite these compelling hypotheses based on fossil evidence, the scarcity of direct proof means it's still possible that romantic love, or at least a precursor to it, predated both bipedalism and altriciality. It might even have originated in a common ancestor of humans and chimpanzees some 5 to 8 million years ago. While chimpanzees primarily engage in opportunistic mating, some of their rarer reproductive strategies exhibit features reminiscent of romantic love, such as mate guarding and more than fleeting associations. One assumption often made in these evolutionary discussions is that less sexual dimorphism in body mass, meaning greater similarity between the sexes, is indicative of monogamy. However, this comparative similarity in human body mass only emerged as recently as 500,000 years ago. This suggests a multi-stage evolution of human pair bonding, implying that romantic love could have developed during any of these protracted periods. Helen Fisher's theory further elaborates on this evolutionary journey, proposing that romantic love, distinct from attachment, is a motivational system designed to choose and focus energy on a preferred mating partner. She calls this courtship attraction, a brain system that evolved from mammalian mate choice. In many species, a preferred partner is chosen based on a display of physical traits, like the peacock's tail or other distinctive behaviors. Fisher extends this to humans, including the attraction to personality traits and other complex characteristics in her mate choice theory. While courtship attraction in most species is fleeting, lasting mere minutes, hours, or weeks, intense romantic love in humans can endure far longer. Fisher theorizes that as pair bonding evolved through human history, this mammalian courtship attraction became progressively prolonged and intensified, ultimately giving rise to the profound phenomenon of romantic love we experience today. However, not all theories align perfectly. Adam Bode offers a critique of Fisher's courtship attraction model, suggesting it might only encompass initial infatuation or a crush, rather than the core components of romantic love, which include intense attraction, obsessive thoughts, and attachment. A study on love at first sight found that while individuals retrospectively recall such experiences as resembling passionate love, those reporting it currently after a first meeting only showed neutral scores on measures of romantic passion. This led researchers to conclude that love at first sight is more accurately a strong initial attraction, rather than the fully fledged state of being in love. Bode argues this aligns more closely with courtship attraction and should be considered a separate system from the deeper, core components of romantic love. Courtship attraction, he suggests, may involve dopamine, oxytocin, and opioid activity, but it remains an area ripe for further, targeted investigation. Another powerful evolutionary theory, one gaining increasing traction, is the co-option of mother-infant bonding. Co-option, or exaptation, is an evolutionary process where an existing trait is repurposed for a new function. Think of how some fish species co-opted their gas bladders, originally for buoyancy, to produce sound. This process allows for rapid evolutionary adaptation because the underlying structure already exists, needing only modification for a new role. Adam Bode has proposed that romantic love is a suite of adaptations and byproducts, consisting of several interrelated systems, with a significant number evolving by co-opting the mechanisms of mother-infant bonding, specifically the attraction for bonding, obsessive thinking, and attachment. This theory suggests that the genes regulating the profound bond between a mother and her infant were duplicated and then repurposed, taking on a new function in adult pair bonding. Courtship attraction and sexual desire are seen as causally linked adjuncts that were not co-opted but combined and modified within the broader framework of romantic love. This theory is supported by human evidence and, significantly, by research on prairie voles, a species known for its monogamous pair bonds, which show that adult pair bonding utilizes the same neurobiological mechanisms as mother-infant bonding. The parallel between romantic love and the mother-infant dyad has been recognized in academic literature since the 1980s. Attachment theorists like Cindy Hazan and Philip Shaver believe the two shared a common biological process. In 1999, James Leckman and Linda Mays meticulously compared features of romantic love and early parental love, finding substantial similarities. Both are altered mental states marked by preoccupations, exclusivity of focus, a longing for reciprocity, and the idealization of the other. Their trajectories also share uncanny resemblances. Preoccupation intensifies during courtship for romantic love and around childbirth for parental love, then diminishes after a relationship is established or after the postpartum period. Even the use of baby talk by romantic lovers is an echo of this deep, shared origin. In 2004, Andreas Bartels and Samir Zeki conducted the first fMRI study comparing romantic love and maternal love. Their findings were striking. They observed precise overlaps in specific brain areas, including parts of the striatum, the putamen, globus pallidus, and caudate nucleus, and some overlap in the ventral tegmental area. These are crucial areas rich in dopamine and oxytocin receptors. While each type of love also showed unique activations, maternal love, for example, activated the periaqueductal gray matter, associated with pain suppression during intense emotional experiences like childbirth. The shared neural architecture strongly supports the co-option theory. More recent meta-analyses of fMRI experiments have further reinforced these similarities, showing both types of love associated with the left ventral tegmental area, linked to the pleasurable liking aspect of reward, with romantic love uniquely involving the right ventral tegmental area, more associated with reward wanting. In 2003, Lisa Diamond expanded on this, suggesting that adult pair bonding is an exaptation of the affectional bond between infants and caregivers. This powerfully explains phenomena like romantic friendships and platonic infatuations, romantic passion devoid of sexual desire. Dorothy Tenev's study of limerence, an intense, involuntary state of infatuation, often for an unreachable person, reported instances of younger women, identifying as heterosexual, developing such reactions towards older women. Other historical examples include schoolgirls falling violently in love with each other, and Native American men forming intense, non-sexual bonds. Fisher's theory, positing sexual desire as a separate system from romantic love and attachment, further supports this. Diamond argues that romantic love, without sexual desire, can even transcend sexual orientation because, from an adaptive perspective, a parent wouldn't be limited to bonding only with an opposite-sex child. While sexual desire often sparks romantic love, Diamond suggests that shared time and physical touch can serve as substitutes, and that the connection between romantic love and sexual desire is bidirectional, each capable of triggering the other through shared oxytocin pathways in the brain. Given the evolving understanding of evolutionary theories and the limitations of earlier neurochemical models, Adam Bode has proposed a more nuanced new model of romantic love, moving beyond Fisher's predominant, but now simplified framework. He suggests 5 distinct, yet integrated, systems. First, sexual desire, the drive to initiate and be receptive to sexual activity. This involves a complex interplay of hormones and neurotransmitters, including testosterone, dopamine, serotonin, norepinephrine, acetylcholine, histamine, and opioids. Second, courtship attraction, which focuses our energy on a preferred mating partner and promotes courtship behaviors. This can manifest as love at first sight or a crush, and may or may not precede a relationship. Dopamine, oxytocin, and opioids are implicated here. Third, bonding attraction, the foundational attraction for pair bond formation. It's characterized by a strong desire for proximity, separation anxiety, exclusive focus, and a heightened awareness of the loved one. This is powerfully associated with dopamine and oxytocin activity, especially in the ventral tegmental area, and, according to Bode, represents the type of romantic attraction observed in fMRI studies of early-stage romantic love. Fourth, obsessive thinking, the preoccupation or intrusive thoughts about the loved one. This hallmark feature ensures the beloved remains central to our consciousness. And finally, attachment, which is crucial for pair bond maintenance, fostering very close personal relationships. Its psychological features include a heightened sense of responsibility, a longing for reciprocity, and a powerful sense of empathy. Oxytocin, dopamine, opioid activity, and some evidence for vasopressin are all involved in this profound connection. Bode's model suggests that the systems of bonding attraction, obsessive thinking, and attachment, the three systems likely co-opted from mother-infant bonding, together form the core of romantic love, its necessary components. However, all five systems merge into the singular, complex phenomenon we call romantic love, with diverse outcomes depending on individual circumstances and experiences. The exploration of love's ancient code reveals a profound truth. What we experience as an intensely personal, often inexplicable emotion is in fact a deeply ingrained biological imperative, sculpted by millions of years of evolution. It's a masterful design, ensuring our survival, our reproduction, and the flourishing of our species. As we move forward, we will delve deeper into the intricate mechanics of this evolutionary blueprint, exploring the neurochemical dances that ignite attraction, the reward pathways that create addiction, and the hormonal symphonies that forge enduring bonds. Join me as we uncover the thrilling science behind why love feels the way it does, and how our brains are wired for this most extraordinary journey. We've journeyed through the unseen architecture of love and glimpsed its ancient, evolutionary blueprints. Now, let's delve deeper into the intricate dance between our minds and our hearts, exploring why, for many, love feels less like a gentle current and more like an irresistible, all-consuming tide. We're talking about dopamine, its profound embrace, and the striking parallels between falling in love and the grip of addiction. The biological study of romantic love, while still in its relative infancy, with a mere 42 biological studies as of 2021, is rapidly unveiling a captivating world of neurochemicals and hormonal cascades. This growing body of research is beginning to unravel how brain systems, working in concert, orchestrate the complex psychological experience and behaviors we associate with romantic love. Let's be clear, defining romantic love itself has been a shifting target throughout history. From the poetic ideals of courtly love to its modern, more scientific interpretations, the term has evolved. However, from a biological perspective, romantic love is increasingly understood as a motivational state, often tied to a desire for long-term mating with a specific individual. It's a phenomenon that transcends age, manifesting with distinctive cognitive, emotional, behavioral, social, genetic, neural, and endocrine activity in both sexes. Ultimately, it serves critical functions throughout much of our lives: mate choice, courtship, sex, and pair bonding. It's a complex suite of adaptations and byproducts, a testament to our recent evolutionary journey. Crucially, this biological definition highlights that romantic love isn't necessarily a dyadic, social, or interpersonal experience from the outset. You see, romantic love can be felt even outside the context of an established relationship. Consider unrequited love, where feelings aren't reciprocated, yet the intensity remains. A person can develop these profound emotions for a potential partner before any tangible relationship has even begun, sometimes for someone they barely know, or not at all, as in the phenomenon of love at first sight, or even parasocial attachments. This early, often obsessive and addictive stage of romantic love has many names: being in love, passionate love, infatuation, limerence. The terminology is still fluid, but the underlying experience is universally recognized. This initial, intense phase is also distinct from the attachment system that theorists like John Bowlby described. While attachment is critical for long-term bonding, early-stage romantic love is thought to engage additional, distinct brain systems with their own unique evolutionary histories. It's also important to differentiate romantic love from mere sexual attraction, though they most often occur hand in hand. The expression of romantic love varies wildly. One cross-cultural study of individuals currently in love identified 4 distinct clusters, each with differing degrees of intensity, obsessive thinking, commitment, and even frequency of sexual activity. And while intense romantic love is typically thought to be time-limited, often lasting 12 to 18 months or up to 3 years, a rare phenomenon known as long-term intense romantic love sees some individuals maintain that initial passionate attraction for a decade or even longer, albeit with fewer of the obsessive features. Now, let's explore the idea of independent emotion systems. Pioneering researchers like Helen Fisher and her colleagues have proposed that the brain systems involved in mammalian reproduction can be broken down into at least 3 distinct parts. Neuroscientists generally agree that basic emotions emerge from specialized circuits of neural activity, many of which we share with other mammals. These ancient brain systems evolved to direct fundamental behaviors. Among them, it's hypothesized, are at least 3 discrete, yet interrelated emotion motivation systems in the mammalian brain, designed for mating, reproduction, and parenting. First, there's lust, the primal sex drive, or libido. Second, we have attraction, often referred to as early-stage romantic love, passionate love, or infatuation. This system is associated with feelings of exhilaration, obsessive or intrusive thoughts, and that profound craving for emotional union. And third, attachment, also known as companionate love, which is rooted in the attachment theory. This system is linked to feelings of calm, security, and comfort, but also, tellingly, separation anxiety when apart. In Fisher's model, these systems generally work in concert, yet they can also become disassociated and act independently. Imagine a person in a long-term partnership feeling deep attachment for their spouse, while simultaneously experiencing intense romantic love, attraction for someone else entirely, and perhaps sexual attraction, lust for still others, all at once. This independent emotions theory helps explain why people can fall in love without sexual desire, as seen in platonic infatuation for a friend. Fisher originally associated each system with different neurotransmitters and hormones: lust with estrogen and androgens, attraction with dopamine, norepinephrine, and serotonin, and attachment with oxytocin and vasopressin. However, modern research reveals that these associations aren't as neatly compartmentalized as initially proposed. Romantic love has also been linked to endogenous opioids, cortisol, and nerve growth factor, which weren't part of Fisher's earlier model. Furthermore, while the idea of interrelated systems remains useful, more recent theories suggest that the attachment system is, in fact, active even during early-stage romantic love, alongside the infatuation component. Fisher's original model, while foundational, is now considered somewhat outdated. Now, let's turn our attention to the evolutionary roots of these systems. Evolutionary psychology provides a powerful framework for understanding why these psychological functions exist, emphasizing their adaptive nature and the reproductive benefits they confer. It offers a variety of compelling explanations for romantic love. Romantic love is a profoundly powerful commitment device. It works to suppress the search for alternative mates, sometimes even irrationally so, when a seemingly more desirable partner might appear. This commitment also signals fidelity to the current partner. Furthermore, it can subtly signal to potential alternative mates, disincentivizing them from pursuing you. The emergence of longer pair bonds in human evolution coincided with the fascinating development of concealed ovulation, meaning that, generally speaking, it's impossible to determine when a woman is ovulating. This necessitated partners staying together and engaging in sexual activity throughout the entire menstrual cycle. Commitment, in this context, became highly adaptive, facilitating this continuous bond and, crucially, enabling shared child care. The intense feelings of love might be the brain's psychological reward, a powerful incentive system for successfully navigating the complexities of commitment. The sheer intensity of romantic love and why people often become fools for love can also be illuminated by the handicap principle. This principle, often seen in nature, addresses the contention between honest and fake signaling. When genuine emotions evolved, a niche opened for sham emotions, which are less risky to express. An honest signal, to be truly credible and avoid becoming worthless due to widespread faking, must be costly to produce. Think of the peacock's tail, a cumbersome, nutrient-consuming display. Only a healthy peacock can afford such an extravagant handicap, making it an honest indicator of fitness to potential mates. Romantic love, in its bewildering, besotting intensity, might have evolved as a similar handicap, a commitment so profound and costly, like handcuffing oneself to railroad tracks, that it serves as undeniable proof of one's genuine intentions. Romantic love may have even evolved to override pure rationality, compelling us to reproduce despite the considerable costs and sacrifices of raising a child, or even against a rational will to remain single or child-free. It's a powerful, primal driver. Furthermore, romantic love signals parental investment. Paternal investment, facilitated by pair bonds, is strongly linked to better outcomes for children, from infancy through later development. Children raised within pair bonds tend to be more socially competitive and have a higher likelihood of surviving to reproductive age. Being in love can also spark creativity, suggesting romantic love may have evolved as a sophisticated courtship display. Art, music, and literature, in this view, serve a function akin to a peacock's tail, but as a display of mental prowess designed to impress and captivate a potential partner. Many authors believe creativity is an especially potent element of the male courtship display. Romantic love can also conserve valuable time and metabolic energy by focusing courtship efforts on a single individual, rather than diffusing them across many. And perhaps most importantly, successful pair bonding predicts better health and survival. Happy, well-functioning romantic relationships contribute significantly to both mental and physical well-being, especially when navigating stress. Conversely, the dissolution of a pair bond, such as divorce, is associated with increased vulnerability to disease, depression, substance abuse, and negative outcomes for children. For instance, heart attack victims living alone are statistically more likely to suffer a second attack. Monogamous pair bonding also plays a role in preventing sexually transmitted diseases, which can severely compromise fertility, particularly for women. Certain STDs, like syphilis, increase the risk of miscarriage and can harm an unborn child. The strongest predictor of contracting an STD is the number of sexual partners, making limited partnerships a vital strategy for protecting reproductive health. Finally, romantic love promotes exclusivity through mate guarding. Romantic jealousy, a frequent companion to being in love, evolved as a protective mechanism against the threat of losing one's beloved to a rival. When it motivates us to maintain a healthy relationship, jealousy is adaptive up to a point. However, it can also devolve into pathological jealousy, where delusional beliefs about a partner's infidelity persist, regardless of evidence. The exact timing of romantic love's evolution remains a subject of intense scientific inquiry. Modern romantic love is generally thought to have emerged either during or after the development of bipedalism, the ability to walk upright. The earliest hominid with substantial evidence of bipedalism and some indications of pair bonding is Ardipithecus ramidus, dating back approximately 4.4 million years. It's been proposed that monogamous pair bonding, a rare trait among mammals, evolved during this period because bipedal mothers, needing to carry infants in their arms or on their hips rather than on their backs, became more vulnerable. This vulnerability would have necessitated additional male support for food and protection, the emergence of what we might call husbands or fathers. Another proposed selection pressure is the evolution of infant altriciality, the state of being born immature and helpless, and the development of a large brain size at birth, which occurred around 2 million years ago. At this critical juncture, the infant's fully developed head became too large to pass through the mother's pelvic birth canal, leading to premature births and underdeveloped infants compared to other species. This also placed a greater burden on mothers, making paternal support even more invaluable. Given the scarcity of definitive evidence, it's also plausible that romantic love, or a precursor to it, predated bipedalism and altriciality, perhaps originating in a common ancestor of humans and chimpanzees 5 to 8 million years ago. While chimpanzees primarily mate opportunistically, some of their rarer reproductive strategies display features reminiscent of romantic love, involving mate guarding and more than fleeting associations. One assumption and hypothesis based on fossil evidence is that less sexual dimorphism in body mass, meaning greater similarity between sexes, is indicative of monogamy. However, this comparative similarity in human body mass only emerged as recently as 500,000 years ago. This suggests that human pair bonding likely evolved through multiple stages, and romantic love could have emerged during any of these periods. Helen Fisher's theory posits that romantic love, which she distinguishes from attachment, is a motivation system designed for choosing a preferred mating partner and focusing energy on that individual. She believes this brain system evolved from mammalian mate choice, also known as courtship attraction. This phenomenon involves selecting a preferred mate based on displays of physical traits, like a peacock's tail or specific behaviors. Fisher expands this to humans, including attraction to personality traits and other characteristics in her mate choice theory. Courtship attraction shares behavioral similarities with human romantic love, and both activate dopaminergic reward circuits. In most species, courtship attraction is fleeting, lasting minutes, hours, days, or weeks. However, in humans, intense romantic love can endure much longer. Fisher suggests that over the course of human evolution, mammalian courtship attraction may have become prolonged and intensified as pair bonding evolved, eventually blossoming into the phenomenon we know as romantic love today. However, a critique of Fisher's theory by Adam Bode argues that courtship attraction primarily encompasses love at first sight, or a crush, and that the core components of romantic love, including intense attraction, obsessive thoughts, and attachment, actually evolved through a process called co-option of mother-infant bonding. A study on love at first sight found that while people retrospectively recall features resembling passionate love, those currently experiencing it immediately after meeting someone reported only neutral scores on romantic love measures. This suggests that the remembered account of falling in love at first sight with intense passion may often be a memory confabulation. Furthermore, the study found that love at first sight was strongly related to the potential partner's physical attractiveness, leading researchers to conclude it's more akin to a strong initial attraction than a full state of being in love. Bode argues this aligns more closely with courtship attraction and should be considered a separate system from core romantic love. Courtship attraction may involve dopamine, oxytocin, and opioid activity, but more research is needed to fully understand it. The concept of co-option is fascinating. It's an evolutionary process where an existing trait is repurposed to serve a new function. Think of how some fish species co-opted their gas bladders to produce sound. Co-opted traits can be morphological or behavioral, allowing for rapid evolutionary adaptation, seemingly faster than traditional Darwinian explanations might suggest. A seemingly new trait can emerge quickly because its underlying structure already existed, only needing modification to function in a new way. Adam Bode has proposed that romantic love is a suite of adaptations and byproducts, comprising several interrelated systems, many of which evolved by co-opting aspects of mother-infant bonding, specifically attraction for bonding, obsessive thinking, and attachment. This co-option theory suggests that the genes regulating mother-infant bonding were duplicated and then repurposed for a new function. Courtship attraction and sexual desire, while causally linked adjuncts, were not co-opted but rather combined and modified within the framework of romantic love. This theory is supported by human evidence and research on prairie voles, which demonstrates that pair bonding utilizes the same mechanisms as mother-infant bonding. Academic literature has drawn parallels between romantic love and the mother-infant dyad since the 1980s. Attachment theorists like Cindy Hazan and Philip Shaver believed they shared a common biological process. In 1999, James Leckman and Linda Mays extensively compared features of romantic and early parental love, finding substantial similarities. Both are altered mental states characterized by preoccupations, exclusivity of focus, a longing for reciprocity, and idealization of the other. Their trajectories also mirror each other. Preoccupation increases during courtship for romantic love and around birth for parental love, then diminishes once a relationship is established or shortly after the postpartum period. Even the uncanny similarity of baby talk between romantic lovers is observed. In 2004, Andreas Bartels and Samir Zeki were the first to use fMRI to compare romantic and maternal love, focusing on brain areas rich in oxytocin and vasopressin receptors. They found precise overlap in specific regions, including parts of the striatum, putamen, globus pallidus, and caudate nucleus, and some overlap in the ventral tegmental area, VA, areas with dopamine and oxytocin receptors. Each type of love also showed unique activations. Notably, maternal love activated the periaqueductal gray matter, an area linked to endogenous pain suppression during intense emotional experiences like childbirth. Two meta-analyses of fMRI experiments have also confirmed these similarities between maternal and romantic love. A 2022 meta-analysis by Shi et al. Found that both types of love activated the left ventral tegmental area, associated with the pleasurable aspect of reward or liking, while romantic love additionally engaged the right ventral tegmental area, more associated with reward, wanting. In 2003, Lisa Diamond suggested that adult pair bonding is an exaptation of the affectional bond between infants and caregivers. This helps explain phenomena like romantic friendships, platonic infatuations, or passionate love without sexual desire. Examples include instances of limerence, love madness for an unreachable person, where a woman identifying as heterosexual might develop intense feelings for an older woman, or historical accounts of schoolgirls falling violently in love with each other. Diamond argues that romantic love, without sexual desire, can even transcend sexual orientation because it wouldn't have been adaptive for a parent to only bond with an opposite-sex child. While sexual desire often sparks romantic love, Diamond suggests that time spent together and physical touch can serve as substitutes. She believes the connection between romantic love and sexual desire is bidirectional, with each capable of causing the other due to shared oxytocin pathways in the brain. Given these evolving theories and the acknowledged simplification of Fisher's earlier neurochemical model, Bode has proposed a new, more nuanced model involving 5 distinct systems. Sexual desire * the drive to initiate and be receptive to sexual activity, implicated with various neurotransmitters like testosterone, dopamine, serotonin, norepinephrine, acetylcholine, histamine, and opioids. Courtship attraction * for choosing and focusing energy on a preferred mating partner, promoting courtship behaviors. This includes love at first sight, or a crush, and may be intertwined with other forms of attraction, though it might not always precede a relationship. It could be associated with dopamine, oxytocin, and opioids. Bonding attraction * the core attraction for pair bond formation, characterized by a strong desire for proximity, separation anxiety, exclusivity of focus, and heightened awareness of the loved one. This is linked to dopamine and oxytocin activity, particularly in the ventral tegmental area, and is the type of romantic attraction observed in fMRI experiments of early-stage romantic love. Obsessive thinking * preoccupation or intrusive thoughts about the loved one. Some authors have drawn parallels to obsessive-compulsive disorder, though evidence is limited and ambiguous. Attachment * for pair bond maintenance and very close personal relationships, featuring a heightened sense of responsibility, a longing for reciprocity, and a powerful sense of empathy. This system is associated with oxytocin, dopamine, and opioid activity, with some evidence for vasopressin's involvement. Bode suggests that bonding attraction, obsessive thinking, and attachment, the 3 systems co-opted for mother-infant bonding, form the necessary core of romantic love. However, all 5 systems merge into a single, complex phenomenon of romantic love, with outcomes varying based on circumstances. Now, let's turn our attention to the mechanics, specifically the profound connections between reward, motivation, and addiction. The early stage of romantic love is frequently compared to a behavioral addiction, with the substance being the beloved person. Addiction involves a phenomenon known as incentive salience, often called wanting. This is the property by which environmental cues become attention-grabbing, attractive, and act like a motivational magnet, pulling a person toward a specific reward. Incentive salience differs from conscious craving. It can motivate behavior unconsciously. In the incentive sensitization theory of addiction, repeated drug use makes the brain hypersensitive to drugs and drug cues, leading to pathological levels of wanting those drugs. People in love are thought to experience a similar incentive salience in response to their beloved. Lovers also share other striking similarities with addicts: tolerance, dependence, withdrawal, relapse, craving, and mood modification. Incentive salience is largely mediated by dopamine projections within the brain's mesocorticolimbic pathway, a region vital for reward, motivation, and reinforcement learning. Dopamine signaling for incentive salience originates in the ventral tegmental area, the VTA, one of 2 primary brain areas producing dopamine. From the VTA, projections extend to areas like the nucleus accumbens, the NAC, in the ventral striatum. Here, dopamine activity attaches motivational significance to stimuli associated with rewards. Brain scans of people in love, often using fMRI while they view a photograph of their beloved, show distinct activations in these very areas, the VTA and NAC. Another dopamine-rich area of the reward system active in romantic love is the caudate nucleus, located in the dorsal striatum. This region, containing 80% of the brain's dopamine receptor sites, is implicated in reinforcement learning and has shown activity in response to monetary rewards and even cocaine. This consistent activity in reward and motivation areas suggests that early-stage, intense romantic love is fundamentally a motivation system, a goal-oriented state, rather than a specific emotion. This aligns perfectly with the description of romantic love as a desire or longing for union with another person and strongly supports its resemblance to addiction. In addiction research, a crucial distinction is drawn between wanting a reward, incentive salience, tied to mesocorticolimbic dopamine, and liking a reward, pure pleasure, linked to hedonic hotspots. These 2 aspects are entirely dissociable. Individuals can be addicted to drugs, compulsively seeking them out even when the drug no longer produces a high or when the addiction severely damages their lives. They can want something, feel compelled towards it due to incentive salience, even when they do not consciously wish for it. In a similar vein, people in love may want a beloved person even when interactions with them are not pleasurable. Imagine wanting to contact an ex-partner after a rejection, knowing the experience will only bring pain. It's also possible for someone to be in love with a person they don't actually like or who treats them poorly. Academics have proposed various theories for how addictions begin and perpetuate. One prominent theory, developed by Wolfram Schultz, involves a dopamine signal that encodes a reward prediction error, or RPE. This RPE is the difference between the predicted value of a reward from an action and the actual value received. Did the reward exceed, meet, or fall short of expectations? In this theory, dopamine also plays a role in reinforcement learning, associating rewards with the cues that predicted them. Abusive drugs like cocaine hijack this mechanism by artificially overstimulating dopamine neurons, mimicking an RPE signal far stronger than anything naturally produced. An alternative model by Kent Berridge and Terry Robinson suggests that dopamine signaling causes the motivational output, incentive salience, which is proportional to RPE, but that the dopamine signal itself might be an effect of learning rather than its direct cause. However, there's overwhelming evidence that dopamine guides both learning and motivation. The computation of dopamine signaling is complex, receiving inputs from various brain areas, though its output, primarily from the VTA, is a relatively homogeneous signal encoding the level of RPE. One study investigated whether individuals in long-term romantic relationships experienced RPE in response to having their partner's appraisal of them validated or violated, finding that they did. This study used fMRI to reveal that reward areas like the VTA and striatum responded in a manner consistent with other RPE research. Most fMRI studies of romantic love have focused on participants viewing a photograph of their beloved, and the resulting reward system activity has been interpreted in terms of salience. It's important to note that research has not yet fully explored whether romantic love shares all neurobiological aspects of addiction. Despite the similarities, there are also significant differences. One major distinction is that the trajectories diverge. The addictive aspects of romantic love tend to diminish over time within a relationship. In contrast, in drug addiction, the detrimental aspects magnify over time with repeated use, escalating into compulsions, loss of control, and a pervasive negative emotional state. It's been speculated that this divergence could be linked to the presence of oxytocin activity in romantic love, which is absent in drug addiction. Oxytocin appears to ameliorate the effects of drug withdrawal and may inhibit the more long-term, excessive impacts of addiction. Academics are not in universal agreement on whether love is always an addiction or when it requires treatment. The term love addiction has had an amorphous definition over the years and is not yet a recognized psychiatric condition. However, a recent definition suggests that individuals addicted to love tend to experience negative moods and effects when away from their partners and have the strong urge and craving to see their partner as a way of coping with stressful situations. Other authors extend the definition to include rejected lovers or specify that love becomes an addiction when it involves abnormal processes with negative consequences. A broader perspective views all love as an addiction or simply an appetite, akin to our fundamental dependence on food. Research on behavioral addictions is less extensive than that on substance abuse, but a growing body of evidence indicates that some individuals are susceptible to showing brain patterns similar to drug addicts in response to natural rewards like food and sex, particularly in the case of gambling addiction. Romantic love might be a natural addiction, distinct from drug addiction in its prosocial nature and its evolutionary purpose of facilitating pair bonding. Helen Fisher, Arthur Aaron, and their colleagues have proposed that romantic love is a positive addiction when reciprocated and beneficial, but a negative addiction when unrequited or inappropriate. This intricate dance between our neurobiology and our deepest desires continues to unfold, revealing the profound and often startling connections between love and the very mechanisms that drive our most powerful cravings. Welcome back, Cosmic Explorers, to another deep dive into the most profound and sometimes perplexing aspects of our universe. Last time, we navigated the intricate dance of dopamine, unraveling why the intoxicating rush of new love often mirrors the powerful grip of addiction. We saw how this essential neurochemical fuels our desire, ignites our obsession, and propels us towards connection. But dopamine, as powerful as it is just one instrument in the vast, complex orchestra that plays the symphony of human connection. Tonight, we delve deeper, exploring the harmonious interplay of other crucial players, the hormones, the endogenous opioids, and the intricate neural pathways that collectively weave the very fabric of attachment, comfort, and the enduring bond we call love. This is the symphony of connection, where chemistry becomes feeling and biology blossoms into belonging. The scientific exploration of romantic love, particularly its biological underpinnings, is, in many ways, still in its infancy. For all our advanced understanding of the cosmos, the deepest oceans, and the furthest reaches of the quantum realm, the inner universe of human emotion remains a frontier ripe for discovery. Consider this. As of just a few years ago, the total number of biological studies on romantic love barely exceeded 4 dozen. This scarcity of research isn't a testament to the insignificance of love, but rather to its immense complexity, its elusive nature, and the sheer difficulty of capturing something so vast and subjective within the confines of a laboratory. For centuries, the concept of romantic love itself has been a shifting tapestry, its definition molded by poets, philosophers, and cultural norms. Initially, it often evoked images of courtly love and idealized, almost unattainable adoration. Yet, in the biological and psychological sciences, particularly in fields like neuroscience and anthropology, the term has begun to coalesce around a more tangible, mechanistic understanding. Here, romantic love isn't merely a poetic ideal, but a discernible brain system, or rather, a collection of systems intimately linked to pair bonding and mating, carrying with it a distinct suite of psychological properties. In 2021, a comprehensive review by Bode and Kushnick sought to synthesize this burgeoning understanding, offering a biological definition of romantic love that grounds this ethereal experience in the tangible world of our bodies and brains. They posited that romantic love is fundamentally a motivational state, typically associated with a desire for long-term mating with a particular individual. It is an experience that transcends age, manifesting throughout our lives and intricately connected to a dazzling array of cognitive, emotional, behavioral, social, genetic, neural, and endocrine activities in both sexes. Its purpose over the vast expanse of human existence has been to facilitate mate choice, courtship, sexual reproduction, and the profound, enduring act of pair bonding. It is in essence a sophisticated suite of adaptations and their byproducts, etched into our very being by the relentless sculptor of evolution. What's fascinating about this biological perspective is its nuanced understanding of romantic love's reach. It's not necessarily a dyadic, social, or strictly interpersonal phenomenon. One can experience the powerful currents of romantic love outside the context of an established relationship. Think of unrequited love, where intense feelings burn brightly without reciprocation, or the almost magnetic pull towards a potential partner, perhaps someone barely known, a fleeting glimpse igniting what we often call love at first sight, or even the deep, one-sided affection of parasocial attachments. These instances highlight that the internal, biological machinery of love can operate independently of a fully formed external bond. The early stages of this experience, often characterized by an almost addictive intensity, are known by many names. Being in love, passionate love, infatuation, limerence, or even obsessive love. The scientific community, still grappling with this new frontier, has yet to settle on a unified lexicon or a consistent set of methodologies, a testament to the profound challenge of studying something so deeply personal and intrinsically subjective. Distinctions are often drawn between this early, passionate phase and the more enduring attachment system theorized by attachment theorists like John Bowlby. While some have argued that attachment theory could encompass all forms of love, modern consensus largely holds that romantic love and attachment, while deeply intertwined, are not identical concepts. Each is believed to involve distinct brain systems with their own unique evolutionary trajectories. Romantic love, importantly, is also separate from mere sexual attraction, though in the grand tapestry of human experience, these two threads are most often found woven together. The expression of romantic love is, of course, as varied as humanity itself. Cross-cultural studies have revealed intriguing clusters of in-love individuals displaying different intensities, varying degrees of obsessive thinking, commitment levels, and even frequencies of sexual activity. Typically, the most intense, fiery phase of romantic love is thought to have a limited lifespan, perhaps 12 to 18 months, or at most 3 years. However, in a rare and captivating phenomenon known as long-term intense romantic love, some individuals report experiencing that initial, fervent attraction for 10 years or even longer. While retaining the powerful draw of early-stage love, these long-term bonds tend to shed the more obsessive features, evolving into a profound and enduring connection. In the intricate dance of our inner world, Helen Fisher and her colleagues proposed a fascinating model, suggesting that the brain systems governing mammalian reproduction and bonding can be parsed into at least 3 distinct, yet interrelated components. Imagine them as separate, yet interacting emotional engines, each firing to drive specific behaviors. First, there is lust, the raw, fundamental sex drive, or libido. This is the primal urge, driven by estrogen and androgens, compelling us towards physical intimacy. It's the ancient call of procreation, ensuring the continuation of our species. Second, we have attraction, or what we often refer to as early-stage romantic love, passionate love, or infatuation. This is the exhilarating, often dizzying state associated with obsessive or intrusive thoughts and an intense craving for emotional union. Here, dopamine, norepinephrine, and serotonin play leading roles, orchestrating the emotional highs and lows, the persistent yearning for the beloved. And third, there is attachment, the deep, abiding bond described by attachment theory, sometimes called companionate love. This system is characterized by feelings of calm, security, and profound comfort in the presence of a loved one, accompanied by distinct separation anxiety when apart. Oxytocin and vasopressin are the key players in this realm, fostering the sense of stability and enduring connection. Fisher's genius lies in recognizing that while these systems often work in concert, they are crucially independent. They can, at times, become disassociated, operating autonomously. Consider a scenario. An individual in a long-term, committed partnership may feel profound attachment to their spouse, a sense of deep comfort and security. Simultaneously, they might experience intense romantic attraction for someone else, a powerful, infatuated longing. And, to complicate matters further, they could still feel a purely sexual lust for yet another individual, all at once. This disassociation helps explain the complex, sometimes contradictory, nature of human relationships. Lisa Diamond further leveraged this independent emotions theory to explain phenomena like platonic infatuation, where a deep, romantic connection can form without any underlying sexual desire. While Fisher's initial model elegantly assigned specific neurotransmitters to each system, modern research reveals a more complex, interwoven tapestry. The associations are not always as clearly defined as once proposed. Beyond estrogen, androgens, dopamine, norepinephrine, serotonin, oxytocin, and vasopressin, romantic love has also been linked to endogenous opioids, cortisol, and nerve growth factor, adding even more layers to this biological symphony. Furthermore, newer theories contend that the attachment system is, in fact, active even in the early, infatuated stages of romantic love, rather than emerging later. While Fisher's model, in its original form, may be considered somewhat outdated in light of new findings, the fundamental concept of these distinct, yet interrelated, systems remains a powerful and useful framework for understanding the intricate biology of love. The very existence of these complex emotional systems begs a fundamental question. How did they evolve? Evolutionary psychology offers compelling frameworks for understanding the why behind these profound psychological functions, grounding them in the adaptive benefits they once conferred. A central tenet of evolutionary psychology is that a given trait must, at some point, have provided a reproductive advantage. From this perspective, romantic love emerges as a remarkably powerful commitment device. Imagine a world where our ancestors needed to form stable pair bonds to raise vulnerable offspring. Romantic love, in its intoxicating intensity, effectively suppresses the search for alternative mates, even to an irrational degree. It signals to a partner, "I am committed to you," and perhaps just as importantly, signals to potential rivals, "This one is taken," thereby disincentivizing competition. The emergence of longer pair bonds in human evolution coincides with the intriguing development of concealed ovulation, where a woman's fertile window is not overtly signaled. This necessitated partners staying together, engaging in sexual activity throughout the menstrual cycle, maximizing reproductive success. Commitment, therefore, was adaptive, crucial for both continuous mating and, vitally, for the demanding task of childcare. Perhaps, then, the exquisite feelings of love are the brain's profound psychological reward for solving the ancient, existential problem of commitment. The sheer, sometimes bewildering intensity of romantic love, the way it can transform us into fools for love, can be illuminated by the handicap principle. This principle posits that honest signaling, particularly in mate selection, must overcome the challenge of deception. When genuine emotions evolved, a niche for sham emotions for superficial displays was created. The way to prove an honest signal is genuine is to make it costly, something that cannot be easily faked. Think of the peacock's magnificent tail, a cumbersome, nutrient-intensive display that only a truly healthy male can afford to grow and maintain. It's a handicap, yes, but precisely because it's a handicap, it's an undeniable indicator of genetic fitness. In a similar vein, the bewitching, almost besotting nature of romantic love, the way it compels individuals to handcuff themselves to railroad tracks metaphorically, might have evolved as a profound handicap, an irrefutable testament to one's genuine, unwavering commitment. Romantic love, some evolutionary theorists propose, may also have evolved to override rationality itself. Raising a child is an enormous, costly endeavor. To ensure reproduction, regardless of the rational will to remain single or child-free, love acts as a powerful, almost primal force, compelling us to embrace the demanding journey of parenthood. Furthermore, romantic love powerfully signals paternal investment. Studies consistently show that paternal involvement, facilitated by strong pair bonds, leads to significantly better outcomes for children, from infancy through adulthood. Children raised in stable pair bonds are more socially competitive, enjoy better health, and are more likely to survive to reproductive age. Love, in this sense, becomes a profound investment in the future of our genetic lineage. The creative surge often experienced by people in love suggests another evolutionary role: romantic love as a courtship display. Art, music, and literature, much like the peacock's tail, can be seen as elaborate displays of mental prowess, designed to impress and captivate a potential partner. Some authors specifically suggest that creativity, in this context, might have evolved as a crucial component of the male courtship display. By focusing courtship efforts on a single individual, romantic love also serves to conserve time and metabolic energy, a pragmatic advantage in the challenging environments of our ancestors. Beyond reproduction and resource allocation, successful pair bonding is intimately linked to better health and survival. Happy, well-functioning romantic relationships are powerful contributors to mental and physical well-being, especially when facing life's inevitable stresses. Conversely, the dissolution of a pair bond, through divorce or separation, is associated with increased vulnerability to disease, depression, substance abuse, and negative outcomes for children. Studies have even shown that victims of heart attacks are more likely to suffer subsequent attacks if they live alone, underscoring the profound protective effect of connection. Finally, in a world where sexually transmitted diseases compromise fertility, particularly for women, monogamous pair bonding offers a crucial defense. The strongest predictor of contracting an STD is the number of sexual partners, and by promoting exclusivity, romantic love serves to limit this risk, safeguarding reproductive health and the well-being of future generations. Jealousy, often an uncomfortable companion to love, is seen in this light as an adaptive mechanism, a vigilant guardian against the threat of losing one's beloved to a rival. While it can devolve into pathological forms, moderate jealousy, when it motivates the maintenance of a relationship, serves an important evolutionary function. Pinpointing the exact moment when romantic love, as we understand it, emerged in human evolution is a fascinating, yet challenging, endeavor. The consensus generally places its evolution either during or after the development of bipedalism. Consider Ardipithecus ramidus, an early hominid from about 4.4 million years ago, showing extensive evidence of bipedalism and some indications of pair bonding. It's been proposed that the rarity of monogamous pair bonding among mammals emerged during this period precisely because walking upright required mothers to carry their infants, leaving their hands occupied and making them more vulnerable. This increased vulnerability would have necessitated additional help and protection from males, hence the need for husbands or fathers. Another significant selection pressure might have been the evolution of infant altriciality, the extreme immaturity and helplessness of human infants at birth, coupled with the dramatic increase in brain size around 2 million years ago. Human infants are born profoundly underdeveloped compared to other species because a fully developed infant's head simply couldn't pass through the mother's narrow pelvic birth canal, what's known as the obstetrical dilemma. This early birth placed an immense burden on mothers, making paternal support even more invaluable for the survival and thriving of offspring. However, the scarcity of fossil evidence leaves room for other possibilities. It's plausible that romantic love, or at least a precursor to it, predated both bipedalism and altriciality, potentially originating in a common ancestor of humans and chimpanzees some 5 to 8 million years ago. While chimpanzees are largely opportunistic in their mating, some of their rarer reproductive strategies exhibit features reminiscent of romantic love, including mate guarding and more enduring associations. One assumption in these hypotheses, based on fossil evidence, is that reduced sexual dimorphism in body mass, the similarity in size between males and females, indicates monogamy. Yet, the comparative similarity in human body mass only fully emerged around 500,000 years ago. This suggests that the evolution of human pair bonding likely involved multiple stages, and romantic love could have developed during any of these periods, a testament to its multifaceted origins. Helen Fisher's theory offers a compelling narrative for the evolution of romantic love, positioning it as a motivation system designed to choose and focus energy on a preferred mating partner. She refers to this as courtship attraction, a phenomenon observed across the mammalian kingdom, where individuals select mates based on various displays, from a peacock's vibrant tail feathers to complex behavioral rituals. In humans, Fisher extends this to include attraction to personality traits and other non-physical characteristics. The behaviors associated with courtship attraction share striking similarities with human romantic love, particularly in their activation of dopaminergic reward circuits in the brain. While courtship attraction in many species is fleeting, lasting mere minutes, hours, or days, human intense romantic love has the capacity to endure much longer. Fisher postulates that over the course of human evolution, this mammalian courtship attraction may have become prolonged and intensified as pair bonding became more crucial, eventually blossoming into the profound phenomenon of romantic love we experience today. However, a critique by Adam Bode suggests a more nuanced picture. Bode argues that Fisher's courtship attraction primarily encompasses love at first sight, or a fleeting crush, while the core components of romantic love, the intense attraction, obsessive thoughts, and deep attachment, may have evolved through a process known as co-option of mother-infant bonding. He points to studies on love at first sight, where individuals, when retrospectively recalling the experience, might embellish it with features of passionate love. Yet, when experiencing it in the moment, they report only neutral scores on romantic love measures. Furthermore, these love at first sight experiences often correlate strongly with the physical attractiveness of the potential partner, suggesting it's more akin to a powerful initial attraction rather than the deep, complex state of being in love. Bode argues that this initial, superficial attraction, or courtship attraction, might be a separate system, potentially driven by dopamine, oxytocin, and opioids, but distinct from the deeper, more enduring core of romantic love. Co-option is a fascinating evolutionary process where a pre-existing trait or mechanism is repurposed to serve a new function. Think of a fish's gas bladder, which initially evolved for buoyancy, but in some species was co-opted to produce sound. This process allows for rapid evolutionary adaptation, as the underlying structure is already in place, merely needing modification for a new purpose. Bode proposes that romantic love is a suite of adaptations and byproducts, where several key systems, including the mechanisms for bonding attraction, obsessive thinking, and attachment, were effectively co-opted from the ancient, fundamental system of mother-infant bonding. In this theory, the genes regulating the powerful bond between a mother and her infant were duplicated or modified to serve a new, yet equally vital, function, forming strong, enduring bonds between adults. Courtship attraction and sexual desire, while intertwined, are seen as causally linked adjuncts that were not directly co-opted, but rather combined and modified within this newly formed framework of romantic love. This theory is supported by extensive research on prairie voles, a monogamous species whose pair bonding mechanisms remarkably mirror those of mother-infant bonding. The parallel between romantic love and the mother-infant dyad is not a new idea. Academics have been drawing these connections since the 1980s, with attachment theorists suggesting shared biological processes. In 1999, James Leckman and Linda Mays highlighted substantial similarities between early romantic love and early parental love, noting that both are altered mental states characterized by preoccupations, exclusivity of focus, a deep longing for reciprocity, and an idealization of the other. Even the trajectories of these loves share similarities. Preoccupation increases during courtship for romantic love and around birth for parental love, diminishing once the relationship is established. The uncanny use of baby talk between romantic partners further reinforces this fascinating link. In a groundbreaking 2004 study, Andreas Bartels and Samir Zeki were the first to use fMRI to directly compare romantic love and maternal love. Their findings were remarkable, revealing precise overlaps in brain areas known to have high densities of receptors for attachment hormones like oxytocin and vasopressin. Specifically, they found shared activation in parts of the striatum, including the putamen, globus pallidus, and caudate nucleus, and some overlap in the ventral tegmental area, regions rich in both dopamine and oxytocin receptors. This neurobiological convergence provides compelling evidence for the co-option theory. Interestingly, maternal love also uniquely activated the periaqueductal gray matter, an area associated with endogenous pain suppression during intense emotional experiences, such as childbirth, highlighting the unique demands of parental care. Two subsequent meta-analyses of fMRI experiments have further solidified these findings, confirming significant neurobiological similarities between maternal and romantic love. A 2022 meta-analysis by Shi and colleagues, for instance, found both types of love activated the left ventral tegmental area associated with the pleasurable liking aspect of reward, while romantic love also uniquely engaged the right ventral tegmental area, linked to the wanting or motivational aspect of reward. In 2003, Lisa Diamond expanded on this idea, suggesting that adult pair bonding is an exaptation, another term for co-option, of the affectionate bond between infants and caregivers. This perspective helps explain intriguing phenomena like romantic friendships and platonic infatuations, where deep, romantic passion emerges without sexual desire. Dorothy Tinov's study of limerence, or love madness, for an unreachable person, offered compelling examples. She documented cases of heterosexual women developing intense, romantic, yet non-sexual attachments to older women, or schoolgirls falling violently in love with each other, with all the pangs of unrequited attachment and desperate jealousy. Even historical accounts of Native American men forming intense, non-sexual bonds resembling romantic love fit this framework. Diamond argues that because it would not have been adaptive for a parent to only bond with an opposite-sex child, the systems for romantic love and sexual desire must have evolved independently of sexual orientation. While sexual desire often sparks romantic love, Diamond suggests that time spent together and physical touch can serve as potent substitutes, activating shared oxytocin pathways in the brain, creating a bidirectional link where either love or desire can ignite the other. Given the evolving understanding of these complex systems and the contentions surrounding earlier evolutionary theories, Adam Bode has proposed a more comprehensive model, expanding beyond Fisher's earlier, somewhat simplified neurochemical framework. Bode suggests 5 distinct, yet integrated, systems that collectively form the intricate phenomenon of romantic love. First, sexual desire, the fundamental drive to initiate and be receptive to sexual activity. This involves a complex interplay of testosterone, dopamine, serotonin, norepinephrine, acetylcholine, histamine, and opioids. Second, courtship attraction, the system for choosing and focusing energy on a preferred mating partner, promoting courtship behaviors. This can manifest as love at first sight or a simple crush, often intertwined with other forms of attraction, though it doesn't always precede relationship. Dopamine, oxytocin, and opioids are thought to play a role here. Third, bonding attraction, which forms the very essence of pair bond formation. It's characterized by an intense desire for proximity, deep separation anxiety, an almost exclusive focus on the loved one, and a heightened awareness of their presence. This is intimately linked to dopamine and oxytocin activity, particularly in the ventral tegmental area, and, according to Bode, represents the type of romantic attraction observed in fMRI studies of early-stage love. Fourth, obsessive thinking, the often consuming preoccupation or intrusive thoughts about the loved one. While some have drawn parallels to obsessive-compulsive disorder, the neurobiological evidence for this link remains limited and ambiguous. And fifth, attachment, the system crucial for pair bond maintenance and the nurturing of all close personal relationships. It brings with it a heightened sense of responsibility, a longing for reciprocity, and a powerful sense of empathy. Oxytocin, dopamine, and opioid activity are key here, with some evidence also pointing to the involvement of vasopressin. Bode's model suggests that bonding attraction, obsessive thinking, and attachment, the 3 systems potentially co-opted from mother-infant bonding, form the very core of romantic love, its necessary components. However, all 5 systems are ultimately merged into a single, complex phenomenon, resulting in a rich tapestry of outcomes depending on the unique circumstances and individual nuances. Now, let's turn our attention to the fascinating mechanics of this biological symphony, delving into the neural pathways and chemical processes that give rise to the profound experiences of love. The early, intense stage of romantic love is increasingly being compared to a behavioral addiction, with the substance of addiction being, quite simply, the loved person. Addiction, whether to a substance or a behavior, involves a powerful phenomenon called incentive salience, often referred to as wanting. This is the property by which environmental cues associated with a reward become incredibly compelling, attention-grabbing, and act like a motivational magnet, drawing an individual towards that reward. Incentive salience is distinct from craving, as craving is a conscious experience, while incentive salience can operate on a subconscious level, powerfully shaping behavior without our explicit awareness. In the incentive sensitization theory of addiction, repeated exposure to drugs or addictive behaviors sensitizes the brain, making it hypersensitive to the cues associated with the reward, leading to pathological levels of wanting. People deeply in love are thought to experience this same incentive salience in response to their beloved. They share other striking similarities with addicts: tolerance, dependence, withdrawal symptoms, relapse, intense craving, and mood modification. This powerful incentive salience is mediated by dopamine projections within the mesocortical limbic pathway of the brain, a crucial network involved in reward, motivation, and reinforcement learning. Dopamine signaling for incentive salience originates in the ventral tegmental area, VT, one of the brain's primary dopamine-producing regions, and projects to areas like the nucleus accumbens, NAC, in the ventral striatum. In the NAC, dopamine activity imbues stimuli associated with rewards with profound motivational significance. Brain scans of individuals in love, often while viewing a photograph of their beloved, consistently show activations in these very areas, the VTA and NAC. Another dopamine-rich region of the reward system, the caudate nucleus in the dorsal striatum, also shows significant activity. The caudate nucleus, containing a staggering 80% of the brain's dopamine receptor sites, is implicated in reinforcement learning and has shown activity in response to monetary rewards and even cocaine. This consistent activation of reward and motivation areas strongly suggests that early-stage, intense romantic love is not merely a specific emotion, but a powerful motivation system, a goal-oriented state characterized by a deep desire and longing for union with another person, strikingly consistent with its parallels to addiction. In the realm of addiction research, a crucial distinction is made between wanting a reward, incentive salience, tied to mesocortical limbic dopamine, and liking a reward, pleasure, tied to hedonic hotspots. These two aspects are surprisingly dissociable. Individuals can become addicted to drugs, compulsively seeking them out, even when the drug no longer provides a high, or when the addiction is actively detrimental to their lives. They can want something, feel an irresistible compulsion towards it, even if they don't cognitively desire it. In a similar vein, people in love may want their beloved, even when interactions are not pleasurable. A rejected lover, for instance, might desperately want to contact an ex-partner, even knowing the encounter will only bring pain. It's even possible, in some complex scenarios, to be in love with someone you don't actually like or who treats you poorly. Academics have proposed various theories for how addictions begin and persist. One prominent theory, developed by Wolfram Schultz, involves a dopamine signal encoding a reward prediction error, RPE. This RPE is the difference between the predicted value of a reward and its actual value, whether it was better, equal to, or worse than expected. Dopamine, in this model, is a key component of a reinforcement learning mechanism, associating rewards with the cues that predicted them. Abused drugs effectively hijack this system, artificially overstimulating dopamine neurons, creating an RPE signal far more potent than anything naturally produced. While an alternative model by Kent Berridge and Terry Robinson suggests dopamine primarily causes motivational output proportional to RPE, and that the dopamine signal itself might be an effect of learning rather than its direct cause, There is overwhelming evidence that dopamine guides both learning and motivation. The computation of dopamine signaling is incredibly complex, drawing inputs from numerous brain regions. Yet its output, primarily from the VTA, is a relatively homogeneous signal encoding the level of RPE. A study exploring long-term romantic relationships investigated whether individuals experienced RPE in response to having their partner's appraisals of them either validated or violated. The findings indicated they do, with fMRI showing reward areas like the VTA and striatum responding consistently with other RPE research. Most fMRI studies of romantic love, however, have simply involved participants looking at a photograph, with the resulting reward system activity interpreted in terms of sheer salience. It's important to note that research has not yet fully investigated whether romantic love shares all neurobiological aspects of addiction. Despite the striking similarities, crucial differences exist. A major divergence lies in the trajectory. The addictive aspects of romantic love tend to diminish over time as a relationship matures. In contrast, drug addiction often magnifies its detrimental aspects, escalating into compulsions, loss of control, and negative emotional states. It has been speculated that the presence of oxytocin activity in romantic love, largely absent in drug addiction, might explain this difference. Oxytocin appears to ameliorate the effects of drug withdrawal and may inhibit the excessive, long-term effects of addiction. Academics don't universally agree on whether love is always an addiction or when it requires intervention. The term love addiction itself has had a somewhat amorphous definition. While not yet a formal psychiatric condition, a working definition has emerged. Individuals addicted to love tend to experience negative moods and effects when away from their partners and have the strong urge and craving to see their partner as a way of coping with stressful situations. Some researchers extend this to rejected lovers or specify that love becomes an addiction when it involves abnormal processes with negative consequences. A broader view suggests that all love is, in essence, an addiction or simply an appetite, akin to our fundamental dependence on food. While research on behavioral addictions is less extensive than on drug abuse, there's growing evidence that some individuals are susceptible to brain patterns in response to natural rewards like food or sex that resemble those of drug addicts, particularly in gambling addiction. Romantic love, then, might be a natural addiction, distinct from drug addiction in its pro-social nature and its evolved purpose of pair bonding. Helen Fisher, Arthur Aaron, and their colleagues have proposed that romantic love is a positive addiction when requited and harmonious, and a negative addiction when unrequited or dysfunctional. Now, let's introduce another key player in this intricate ballet of connection, oxytocin. Often dubbed the love hormone or cuddle hormone, oxytocin is critically involved in maternal behavior and adult pair bonding. It's synthesized primarily in the hypothalamus and released into the bloodstream via the pituitary gland, where it's been found circulating in individuals in the early stages of romantic love. Crucially, the hypothalamus also projects oxytocin to other vital brain areas, including the ventral tegmental area, VA, nucleus accumbens, NAC, amygdala, and hippocampus. These projections to reward areas like the VTA and NAC are believed to modulate social salience, essentially influencing the level of dopamine activity in response to socially relevant stimuli. This oxytocin signaling in reward pathways may be a significant source of the profound salience we attribute to a loved one. Studies underscore oxytocin's role. A placebo-controlled study found that administering intranasal oxytocin enhanced the perceived facial attractiveness of a romantic partner when viewing their photograph, compared to an unfamiliar face. fMRI scans in this study revealed enhanced brain activity in reward areas like the VTA and NAC. Another fMRI study identified dopamine-rich genetic expression of an oxytocin receptor gene in the left VTA, a region also active in response to facial attractiveness. In humans, circulating oxytocin levels have been linked to higher levels of interaction between partners and even predicted which couples would remain together 6 months later. Anna Makin aptly describes the combination of oxytocin and dopamine as the glue that makes the early stages of a relationship possible. Oxytocin's influence on human behavior is multifaceted and complex. It appears to lower inhibitions to forming new relationships by deactivating the amygdala, a brain region involved in processing fear and anxiety. Released through physical touch, it fosters feelings of comfort and bonding. It also plays a role in sexual behavior, being released during arousal and orgasm. Beyond romantic and parental bonding, oxytocin contributes to interactions with peers and even strangers, facilitating facial recognition and eye contact. While it enhances trust and altruistic behaviors towards in-groups, partners, children, it can paradoxically also fuel aggression towards outgroups, strangers, rivals. Much of our understanding of oxytocin's role in bonding comes from groundbreaking research on monogamous prairie voles, notably by Larry Young. In these fascinating creatures, both oxytocin and dopamine signaling have been demonstrably shown to influence pair bond development. For example, the number of oxytocin receptors in the NAC is directly correlated with how quickly a partner preference forms. Conversely, injecting either a dopamine or oxytocin receptor antagonist, a drug that blocks transmission, directly into the NAC of a prairie vole can prevent the formation of a partner preference altogether. In contemporary models of the brain systems underlying romantic love, this type of salience or bonding attraction is thought to be present throughout the entire duration of romantic love, including its early, intense stages. This challenges earlier theories, such as Helen Fisher's 1998 model, which posited that oxytocin and dopamine activity were distinct, independent systems, with oxytocin's role only becoming prominent in later stages of a relationship. However, oxytocin levels would still fluctuate based on specific situations, such as less regular interaction and physical touch in cases of unrequited love. This variation could help explain some of the maladaptive symptoms of infatuation, such as sleep difficulties, social anxiety, or clammy hands, which may arise when dopaminergic activity is high without the calming, buffering effect of oxytocin from the attachment system. Adding another rich layer to the symphony of connection, modern research is increasingly highlighting the profound importance of endogenous opioids in love and social attachment. Specifically, the beta-endorphin, the body's most potent endogenous opioid, and the mu-opioid receptor system are proving crucial. While opioids are best known as the body's natural painkillers, they also function as complex neurotransmitters, influencing a wide array of systems. Opioid receptors are distributed throughout the brain, including the limbic system, which governs basic emotions, and the neocortex, involved in conscious decision-making. Opioids are intimately linked to the consummatory part of reward, often referred to as liking or pleasure. They are released in specific brain regions known as hedonic hotspots or pleasure centers, located in areas like the nucleus accumbens and the ventral pallidum. This function extends to social reward, the profound pleasure derived from social interactions. The brain opioid theory of social attachment, BOTSA, a long-standing theory originating in the 1980s and 90s from the work of psychiatrist Michael Leibovitz and neuroscientist Jake Panksepp, summarizes this vital connection. For a time, opioids were overshadowed by the excitement surrounding oxytocin research, perhaps due to the difficulty and expense of studying them, requiring sophisticated techniques like PET scans. However, more recently, opioids have re-emerged as key players, connected to a variety of social experiences, including early-stage romantic love and different attachment styles. While the addictive aspects of love are often compared to cocaine or amphetamine addiction, other facets may, surprisingly, resemble an opioid addiction. BOTSA, in its original formulation, posited that in the absence of social relationships, individuals experience comparatively lower levels of endogenous opioids, driving them to seek out social contact. Social interaction then triggers feelings of euphoria and contentment, but individuals must continue this contact to avoid experiencing withdrawal-like symptoms. Leibovitz originally argued that romantic relationships bear striking resemblances to narcotic addiction, and that individual neurochemical differences could even explain why some people struggle with commitment or remain in abusive relationships. Early experiments supporting Botza were primarily animal studies, but in the 2000s, this research has expanded to include human investigations. Animal studies have provided compelling evidence that separation distress is akin to opioid withdrawal. Experiments with chicks, puppies, guinea pigs, rats, sheep, and monkeys have consistently shown that administering morphine reduces distress vocalizations when separated from their mothers, while administering naloxone, an opioid antagonist, increases them, even in the presence of other members of the same species. In a particularly telling study, mutant mouse pups lacking the mu-opioid receptor gene vocalized less frequently in response to isolation and showed no reduction in distress when given morphine, unlike normal mice. Furthermore, these knockout mice exhibited a reduced preference for their mother's odor, a preference normally mediated by the endogenous opioid system. In non-human primates, endogenous opioids appear to be the source of the euphoria experienced during dyadic social grooming behaviors. Other animal studies link endogenous opioids to the desire for rough-and-tumble play, a physically and socially bonding behavior. In humans, engaging in physical activity with a social element, like rowing, dancing, or laughing, increased pain tolerance more significantly when the activities were synchronized with other people, suggesting an opioid-mediated social reward. A 2010 fMRI experiment provided fascinating human evidence, investigating whether viewing a picture of a romantic partner could reduce pain sensitivity and identifying the activated brain regions. Participants exposed to high temperatures inducing moderate to high pain viewed images of their intensely loved partner, a friend, or engaged in a word association task. Both viewing a romantic partner and the distraction task, but not viewing a friend, significantly reduced pain levels. The fMRI scans revealed that viewing a romantic partner activated specific reward circuits, while the distraction task engaged distinct areas, suggesting two different mechanisms for pain relief. Crucially, some brain areas associated with sensory pain processing showed decreased activity when viewing a romantic partner. An earlier experiment had also shown that viewing photos of a romantic partner reduced experimental pain, though without brain imaging. A 2016 PET scan experiment explored whether non-sexual social touching between romantic partners was mediated by endogenous opioid activity. While social touch did have an effect and was rated as pleasurable, it unexpectedly decreased opioid activity in the brain rather than increasing it. This stands in contrast to previous PET research linking pleasant effect to increased opioid activity. One possible explanation is that touching reduces stress, which in turn might decrease ongoing opioid activity related to distress and pain. This finding, somewhat at odds with primate studies on grooming, suggests there might be species-specific variations in how opioids are involved in social reward. More modern human studies are now exploring blood plasma levels, genetics, and the effects of drugs like morphine and naltrexone on social perception and behavior. Another hallmark, or even a cardinal trait, of romantic love is obsessive thinking about the loved one. This ensures the beloved remains central in one's consciousness. Reports suggest that people can spend as much as 85 to 100% of their days and nights fixated on their love object. One study found that, on average, individuals in love spent 65% of their waking hours thinking about their beloved. Another cluster analysis identified different groups of lovers, with the least intense spending 35% of their time, and the most intense, a staggering 72%. Since the late 1990s, these obsessional features have been compared to obsessive-compulsive disorder, OCD, sometimes paired with the theory that obsessive thinking is linked to lowered serotonin levels during love, though study results on this have been inconsistent or negative. Another theory connects obsessive thinking to addiction, given that drug users also exhibit obsessive thoughts about drug use and accompanying compulsions. In their 1999 theoretical comparison, James Leckman and Linda Mays explored the links between early-stage romantic love, early parental love, and OCD. While their primary aim was to investigate the origins of OCD, their findings shed light on the evolutionary theory of romantic love. Both early romantic love and OCD share features of preoccupation, intrusive thoughts, a heightened sense of responsibility, a need for things to be just right, and certain proximity-seeking behaviors. The authors even speculated that psychasthenia, feelings of incompleteness or imperfection, resembles the longing for reciprocity and idealization inherent in romantic love. Two experiments have explored the relationship between romantic love and serotonin levels using blood samples, assuming peripheral serotonin measures can serve as a marker for central nervous system serotonin. A 1999 study by Donatella Mariziti found that people in love had platelet serotonin transporter, SERT, density similar to unmedicated OCD patients and lower than controls. When retested 12 to 18 months later, SERT density had returned to normal. However, because this study looked at SERT rather than serotonin directly, it's ambiguous whether serotonin levels were actually higher or lower. SERT transports serotonin back into platelets, so a reduction could mean increased plasma levels. A contradictory 2012 study by Sandra Langeslag, which measured blood serotonin levels directly, found differing effects for men and women. Men had lower levels, but women had higher levels, with obsessive thinking in women actually associated with increased serotonin. A 2025 study by Adam Bode further found no association between SSRI use and obsessive thinking or love intensity. Therefore, while romantic love and serotonin are likely associated, the idea that obsessive thinking is solely due to lowered serotonin levels appears inaccurate. Crucially, romantic love isn't a single, monolithic emotion, but rather a powerful motivation or drive that elicits a spectrum of emotions depending on the situation. Its emotional valence is fluid. When things go well, it brings joy, ecstasy, or fulfillment. When things go awry, it can plunge us into sadness, anxiety, or despair. A 2014 study of Iranian young adults found that early romantic love was associated with the brighter side of hypomania, elation, increased mental and physical activity, and positive social interaction, along with better sleep quality. Yet, it also brought stronger symptoms of depression and anxiety, leading the authors to conclude that romantic love is not entirely a joyful and happy period of life. It can be profoundly pleasant or deeply unpleasant, regardless of its intensity. Dorothy Tenev's interview participants eloquently captured this duality. When I felt Barry loved me, I was intensely in love and deliriously happy. When he seemed rejecting, I was still intensely in love, only miserable beyond words. The intensity of love feelings is also distinct from relationship satisfaction. One can be satisfied in a relationship for reasons beyond love for their partner, such as financial security or childcare, or, tragically, be deeply in love with an abuser in an abusive relationship. Unrequited love is a common, yet poorly understood, phenomenon, particularly among young adults. One study found that 63% of respondents had experienced a huge crush at least once in the past 2 years without expressing it, and unrequited love was 4 times more frequent than reciprocated love. Another study reported that 92.8% of participants had experienced at least one powerful or moderate episode of unrequited love in the past 5 years. In 2010, Helen Fisher, Arthur Aaron, and colleagues conducted an fMRI experiment on recently rejected lovers. Participants, on average 63 days post-rejection for relationships lasting about 21 months, reported spending over 85% of their waking hours thinking about their rejecter, experiencing a profound lack of emotional control, and exhibiting unhappiness, sometimes even depression, anger, or paranoia. Similar to studies of happy lovers, scans while viewing a photograph of the rejecting partner showed activations in dopaminergic reward system areas like the ventral tegmental area and nucleus accumbens. Crucially, these activations were even stronger than those observed in happily in-love participants. The activated nucleus accumbens, prefrontal cortex, and orbitofrontal cortex have been linked to assessing gains and losses, while active areas of the insular cortex and anterior cingulate cortex are involved in physical pain and its regulation. This suggests that the intense yearning of unrequited love, despite its pain, still activates the brain's reward circuits, reflecting a persistent wanting even in the face of rejection. In the nascent stages of romantic love, individuals often exhibit a heightened state of hypervigilance, an almost exquisite awareness and sensitivity to their partner's cues, driven by the inherent uncertainty and novelty of a new connection. Over time, as a relationship matures, this hypervigilance can evolve into a profound synchronization. Bonding, it is believed, is partly facilitated by coordinated behaviors that signal reciprocity and by events that evoke eustress, beneficial stress, such as a passionate kiss. The body's stress response system, involving the autonomic nervous system and the hypothalamic pituitary adrenal axis, plays a role here. Experiments measuring cortisol levels have yielded inconsistent results, sometimes showing higher, sometimes lower levels of this stress hormone during early romantic love, suggesting a complex and nuanced involvement of the stress response. The intriguing phenomenon of frustration attraction, sometimes called the Romeo and Juliet effect, suggests that adversity, particularly social or physical barriers, can paradoxically heighten romantic passion. This idea has echoed through history, from the musings of Socrates and Ovid to the ancient Kama Sutra and modern advice columns. Bertrand Russell famously opined that when a man has no difficulty in obtaining a woman, his feeling toward her does not take the form of romantic love. Common barriers include interfering parents, deceptive spouses, or other social customs. Helen Fisher attributes this phenomenon to the mechanics of dopamine. Animal studies have shown that when an anticipated reward is delayed, reward-expecting neurons prolong their firing until the reward is delivered. This sustained neural activity, fueled by anticipation and frustration, may intensify the wanting in human romantic love. Passionate, infatuated love is also thought to thrive in situations involving the uncertainty of intermittent reinforcement. When consummation is withheld, when barriers prevent lovers from meeting regularly, or when perceptions of reciprocation are ambiguous and constantly shifting, love can intensify. This resembles the dynamics of a slot machine, where unpredictable rewards, rather than regular payouts, can lead to gambling addiction and compulsive behavior. The exhilarating high of unexpected wins, the inability to habituate to the experience, magnifies cue-triggered incentive salience, particularly in gambling and drug addictions. Uncertainty theory, in the context of romantic love, is closely associated with Dorothy Tenev's concept of limerence, an addictive, infatuated love often experienced for an unobtainable person. Tenev observed drastic emotional transitions caused by changes in perception of reciprocation, leading to seeming emotional volatility even in otherwise stable individuals. This effect of uncertainty has also been interpreted as attachment anxiety. Even intermittent maltreatment, often known as trauma bonding, has been linked to intensified romantic passion, including suffering. This, again, is believed to be related to intermittent reinforcement and the constant confirmation or violation of expectations. As Elaine Hatfield eloquently put it, consistency generates little emotion. It is inconsistency that we respond to. What would generate a spark of interest is if our admiring friend suddenly started treating us with contempt, or if our arch enemy started inundating us with kindness. The brain, it seems, is exquisitely tuned to the unpredictable. Finally, we turn to the curious phenomenon of positive illusions. People in love have a remarkable tendency to overemphasize the positive aspects of their loved one or their relationship, while simultaneously overlooking or devaluing negative aspects. This cognitive bias has been called crystallization, idealization, the love is blind bias, putting the loved one on a pedestal, or simply seeing through rose-colored glasses. While some past authors viewed this as a malady, fearing that idealization would lead to disillusionment as a relationship progressed. Significant modern scientific evidence contradicts this. Far from being detrimental, positive illusions actually contribute to relationship satisfaction, long-term well-being, and a decreased risk of relationship dissolution. While the exact mechanism remains elusive, certain brain areas are proposed to be involved. The dopaminergic reward system, areas active in romantic love, may play a role in attributing heightened salience to a loved one's positive characteristics. Reduced activation of the dorsal anterior cingulate cortex, involved in error detection and negative social evaluation, could be an adaptive response to a partner's perceived negative traits. Specific regions of the prefrontal cortex might exert top-down control, suppressing emotional responses to attractive alternatives. All of this information is then funneled to the orbitofrontal cortex, where positive and negative information is weighed, ultimately resulting in a biased, subjectively elevated valuation of the partner. Our exploration of the biological underpinnings of love wouldn't be complete without a look at the technological marvels that allow us to peer into the working brain. Brain imaging techniques, such as functional magnetic resonance imaging, fMRI, and positron emission tomography, PET, have revolutionized our understanding of which brain regions are activated during romantic love. Nearly all of these experiments involve participants looking at a photograph of their beloved during a scan, though the specific procedures, such as the duration of love or the task given during the scan, can vary, explaining some differences in results. In 2000, Andreas Bartels and Samir Zeki at University College London conducted the very first fMRI study of romantic love. Their 17 participants, described as truly, deeply, and madly in love for an average of 2.4 years, were shown photographs of their loved one. Two main areas consistently showed activity. The middle insular cortex, associated with gut feelings or butterflies in the stomach, and part of the anterior cingulate cortex, linked to feelings of euphoria. Other activations included the cerebrum, caudate nucleus, putamen, and cerebellum. A later analysis in 2004 also identified activity in the ventral tegmental area, VA, a key dopamine-producing region. Crucially, the study also revealed significant deactivations in the amygdala, involved in fear and risk detection, and the medial prefrontal cortex, MPFC, which helps us understand others' intentions. These deactivations were interpreted as evidence that love is blind, suggesting that people in love may discount risks and misinterpret intentions, sometimes leading to profound folly. In 2005, a groundbreaking study by Arthur Aaron, Helen Fisher, and their colleagues offered the first fMRI insights into early-stage, intense romantic love. This study, lauded for advancing our scientific understanding of infatuated love, involved 17 participants who had just fallen madly in love for a much shorter average duration of 7.4 months. These individuals were more intensely in love, often spending 85% or more of their waking hours thinking of their beloved. Similar to other studies, viewing a photograph of their loved one activated reward and motivation areas like the VTA and parts of the caudate. Activity was also found in the insular and cingulate cortex, regions involved in emotion. Intriguingly, some areas correlated with the length of the relationship, such as the ventral pallidum, implicated in attachment in prairie voles, and the anterior cingulate, linked to obsessive thinking, cognition, and emotion. This study also differentiated between romantic passion and aesthetic preference, finding that the right VTA was active due to romantic passion, while aesthetically pleasing faces activated the left VTA, associated with liking, pleasure, suggesting the right VTA is more involved in wanting, incentive salience. In 2011, a subsequent study by Su et al. Replicated these findings with Chinese participants. Ortigue et al. Utilized fMRI to explore the subliminal influence of romantic love on motivation, examining how these implicit neural representations might differ from studies where stimuli were consciously perceived. In their experiment, participants were subliminally primed for just 26 milliseconds with either their beloved's name, a friend's name, or a word describing a personal passion, followed by a series of symbols and then a target word. Participants were faster to identify whether the target was a word when primed with a love word or passion word, and this speed correlated with their scores on the passionate love scale. The authors interpreted this as evidence that love priming activates brain motivation systems, rather than simply evoking an emotion. The fMRI scans showed brain regions active for love primes similar to previous experiments, including reward and motivation areas like the VTA and caudate, but with additional activation in the bilateral fusiform gyri and angular gyri, regions involved in integrating abstract representations. This was linked to the self-expansion model of interpersonal relationships, where integrating the characteristics of one's beloved into oneself is a rewarding experience that fosters romantic love. In brain scans of individuals experiencing long-term, intense romantic love, those who profess to be madly in love after 10 years or more together, Bianca Acevedo and her colleagues found that attraction, similar to early-stage love, was associated with dopamine reward center activity, wanting. However, long-term attachment in these individuals was also strongly associated with the globus pallidus, a site for opiate receptors identified as a hedonic hotspot, liking. These long-term lovers also exhibited significantly lower levels of obsession compared to those in the early stages of love, highlighting the evolution of the neural landscape over time. Sandra Laneschlag's fMRI study investigated the crucial role of attention on brain activity related to a loved one. Unlike previous experiments where subjects passively viewed photographs, this study used an oddball task to distinguish between instances where the loved one was either the intended target of attention or a mere distraction. Participants were shown random faces and instructed to watch for either a loved one or a friend, pressing a button if the face was the target. The study revealed that activity in the dorsal striatum, a reward system area, was modulated by whether participants were instructed to pay attention to their loved one. That is, the dorsal striatum showed a stronger response to the loved one than to a friend, but only when the loved one was the designated target. This led the authors to conclude that the dorsal striatum is not activated by beloved-related information per se, but only by beloved-related information that is attended. This activity also tended to be smaller in individuals who had been in love or in a relationship for longer. Given the dorsal striatum's role in reinforcement learning, the increased brain activity was interpreted as reflecting prior reinforcement of social actions, leading infatuated individuals to preferentially attend to their loved one. Interestingly, participants were also more prone to mistakenly press the button when distracted by their loved one than by a friend. Finally, many brain scan experiments across both early-stage and long-term romantic love have consistently shown activity in the posterior cingulate cortex, implicated in autobiographical memory of socially relevant stimuli, such as a partner's name, and in attention. Furthermore, most studies have revealed activity in the hippocampus and parahippocampal gyrus, brain areas vitally involved in learning and memory, underscoring how deeply our loved ones become etched into the very fabric of our personal narratives and neural landscapes. And so, we conclude this segment, having journeyed deeper into the intricate biological orchestra that plays the symphony of human connection. We've seen how dopamine fuels the initial fire, how oxytocin weaves the threads of bonding and comfort, and how endogenous opioids provide the exquisite pleasure of social attachment. We've peeled back the layers of evolutionary purpose, exploring how these systems emerge to solve ancient problems of commitment, survival, and reproduction. And we've peered into the living brain itself, witnessing the subtle dance of neural activity that underpins the profound, sometimes bewildering, experience of love. But our journey into the science of love is not yet complete. In our next and final segment, we will venture into the realm of perception, unraveling how the brain constructs the lover's gaze, exploring the fascinating interplay of illusions, uncertainty, and the neural maps that guide us through the complex landscape of romance. Prepare to have your perceptions, once again, gently expanded. We've journeyed through the unseen architecture of love, decoded its ancient evolutionary blueprints, and felt the exhilarating rush of dopamine's embrace. Now, as our exploration deepens, we turn our gaze to the subtler, yet equally profound, ways our minds perceive and construct the object of our affection. We enter the realm of what I call the lover's gaze, a world where uncertainty fuels desire, where the brain actively constructs illusions, and where romance carves its unique map onto our very being. Think for a moment about perception. We often believe we see the world as it truly is, a clear and objective reality. Yet, when love enters the frame, this objectivity often dissolves, replaced by a kind of benevolent bias. It's a phenomenon that has fascinated thinkers for centuries, encapsulated in phrases like love is blind or the act of seeing through rose-colored glasses. But what exactly is happening within the brain to create these powerful, often unwavering, positive illusions? Academically, we refer to this as a form of cognitive bias. Individuals in love tend to amplify the positive attributes of their beloved, while simultaneously minimizing or even entirely overlooking the less desirable ones. This isn't necessarily a flaw in our perception. In fact, modern scientific evidence increasingly suggests that these positive illusions play a crucial role in the health and longevity of a relationship. They contribute to greater relationship satisfaction, enhance long-term well-being, and even reduce the risk of a relationship dissolving. It seems our brains are actively engaged in a constructive process, building an idealized version of our partner that serves to reinforce our bond. The precise mechanisms behind this intriguing phenomenon are still being actively investigated, but we have some compelling theories. The very dopaminergic reward systems we discussed earlier, those magnificent networks lighting up with wanting and pleasure, are believed to be instrumental here. When these areas are highly active in love, they might be attributing heightened salience, a kind of motivational magnet, to the positive characteristics of our loved one. Essentially, our brain is tagging these positive traits as exceptionally important and rewarding, making them stand out far more than any negatives. Consider, too, the role of inhibition. Our brains are incredibly adept at filtering information. In love, we see a reduction in activity in certain areas, such as the amygdala, a region strongly associated with fear and risk detection. This deactivation might lead to a decreased perception of potential threats or flaws in our partner. Furthermore, the medial prefrontal cortex, involved in understanding and predicting the intentions of others, what we call mentalizing, also shows reduced activity. This could be why, at times, people in love seem to disregard clear warning signs or misinterpret motivations that might otherwise be obvious. It's a captivating illustration of how the brain actively works to sustain the romantic narrative, sometimes even at the cost of objective assessment. But the lover's gaze is not just about idealization. It's also profoundly shaped by the intricate dance of uncertainty. As humans, we are wired to seek patterns, to predict outcomes. Yet, in the nascent stages of love, certainty is often a luxury. There's the intoxicating ambiguity of whether feelings are reciprocated, the tantalizing suspense of when you'll next see them, or the subtle shifts in their demeanor that keep you guessing. This dynamic is beautifully explained by concepts like frustration attraction, sometimes romantically dubbed the Romeo and Juliet effect. The idea here is that adversity or barriers, whether social, physical, or emotional, can actually intensify romantic passion. Socrates, Ovid, and even modern advice columnists have remarked on this phenomenon. The theory posits that when a desired reward, like the affection of a beloved, is anticipated but delayed or uncertain, the brain's reward-expecting neurons prolong their firing. This sustained neurochemical activity, particularly involving dopamine, keeps us engaged, keeps us wanting. It's akin to the intermittent reinforcement found in a slot machine. If the machine paid out predictably, it would quickly lose its thrill. But because the rewards are unpredictable, because there's always that tantalizing possibility of a jackpot, the gambler remains captivated, often leading to addiction. In love, this intermittent reinforcement, this delightful uncertainty, can magnify our wanting, that incentive salience, especially when coupled with the profound emotional stakes involved. We constantly seek confirmation, our emotional states oscillating wildly based on perceived validations or rejections. This can lead to a kind of emotional volatility, even in otherwise stable individuals, as described in studies of limerence, that obsessive, infatuated love, often for an unreachable person. The brain is constantly updating its predictions, and each unexpected positive signal acts as a powerful reinforcement. So, how do we begin to map this complex interplay of idealization and uncertainty onto the brain itself? Early pioneering work, such as the fMRI studies by Andreas Bartels and Samir Zeki, gave us some of the first glimpses into the neural landscape of love. They found activations in areas like the middle insular cortex, which is linked to visceral gut feelings, perhaps the very sensation of butterflies in the stomach. They also observed activity in parts of the anterior cingulate cortex, associated with euphoria, and in deeper brain structures like the caudate, nucleus, and putamen, which, as we've discussed, are rich in dopamine receptors and integral to our reward system. Crucially, these studies also identified deactivations. The amygdala, our brain's alarm system, and the medial prefrontal cortex, involved in social cognition and assessing others' intentions, showed reduced activity when individuals were deeply in love. These deactivations lend tangible neuroscientific support to the idea that love is blind, suggesting that when we are truly smitten, our brains might actively suppress critical assessments and risk evaluations. Later research, particularly by Arthur Aaron, Helen Fisher, and their colleagues, delve into the early, intense stages of romantic love, that dizzying period when people have just fallen madly in love. These studies consistently highlighted the activation of those core reward and motivation areas, the ventral tegmental area and the caudate nucleus. Interestingly, they also found correlations between brain activity and the duration of the relationship, with areas like the ventral pallidum, implicated in attachment, and the anterior cingulate, linked to obsessive thinking, showing specific patterns. This research even differentiated between the wanting of romantic passion, which activated the right ventral tegmental area, and the more generalized liking of aesthetically pleasing faces, which stimulated the left VTA. It seems our brain possesses distinct neural signatures for these nuances of desire. Even more subtly, experiments exploring the subliminal influence of romantic love have shown fascinating results. When participants were briefly, unconsciously exposed to their beloved's name, it activated brain regions similar to those seen in conscious viewing, including the VTA and caudate. But it also engaged areas involved in integrating abstract representations, suggesting that love goes beyond mere emotion or attraction. It fundamentally alters how we perceive and integrate the world, even at an unconscious level. This resonates with the idea that in love, we begin to integrate the characteristics of our beloved into our very sense of self, a profound and rewarding experience that further fuels our romantic feelings. What about long-term, intense romantic love, a rare but powerful phenomenon where individuals remain madly in love for a decade or more? Here, the studies show a continuation of dopamine reward center activity, signaling that wanting is still present. However, attachment in these relationships is strongly associated with activity in the globus pallidus, a region rich in opiate receptors and identified as a hedonic hotspot, the seat of liking or pure pleasure. This suggests that while the initial passionate drive remains, it is complemented by a deep, enduring sense of comfort and contentment. In these enduring loves, the obsessive features of early infatuation tend to diminish, replaced by a profound and consistent sense of well-being. The story of the lover's gaze is a testament to the brain's incredible plasticity and its capacity for imaginative construction. It shows us that love is not merely an emotion we feel, but a powerful motivational state, a dynamic interplay of neurochemicals, cognitive biases, and deeply ingrained evolutionary drives. It's a journey into the self, deeply intertwined with the perception of another. And as we conclude this segment, we are left with the profound realization that the architecture of love, in all its complexity and occasional illusion, is one of the most remarkable and enduring masterpieces of the human brain. And so, our extraordinary journey through the biology of romantic love draws to a close. From the ancient evolutionary whispers that sculpted our capacity for connection, to the intricate neurochemical symphonies playing within our brains, we've witnessed how science illuminates the profound mystery of human affection. This is a universe within us, as vast and awe-inspiring as the one above. I hope this deep dive has offered you new perspectives and perhaps even a moment of calm reflection. If you've enjoyed this exploration, please consider leaving a like, sharing it with someone who might appreciate it, and subscribing to Vibing Science for more journeys into the heart of scientific discovery. What did you find most surprising about love today? And what other fascinating mysteries of our world, or indeed ourselves, would you like to explore next? Until our next shared moment, keep questioning, keep learning, and keep vibing with science.